Good morning, everyone. I'm Art Ellis, Vice President for Research and Graduate Studies at the University of California Office of the President. I'd like to extend a warm welcome to all of you viewing the UC Valley Fever Research webinar today. We're pleased to have all of you from around California joining us. And I understand we also have colleagues at NIH, CDC, University of Arizona, and University of Wisconsin with us as well. University of California is the public research arm of the state of California, and we frequently partner with the state to address research issues of concern to Californians. For example, our office manages statewide research programs addressing HIV/AIDS, breast cancer, and tobacco-related diseases. Through its public health and biomedical research programs in 5 medical centers, UC helps improve health of Californians. Having basic research, clinical research, and community research expertise positions UC to make a significant set of advances in Valley fever research. UC researchers have a long history of contributing to our understanding of this disease and in developing diagnostic and treatment methodologies. From UC Davis's Coxey Serology Lab to UC Merced's Valley Fever Network to the BSL-3 labs available at Davis, Irvine, Los Angeles, San Diego, and San Francisco campuses, UC's existing resources and expertise can be leveraged to increase the impact of the $3 million allocated to Valley fever research at the University of California. Your input through this webinar will help us make the best possible use of this important funding resource. Valley fever affects some of California's most vulnerable populations, such as farm workers who produce our food and construction workers who build our communities. Addressing Valley fever is an opportunity for UC to help prevent and treat this debilitating disease. Our ability to make this important investment in Valley fever research is due in large part to California legislators, and in particular to Assembly Member Rudy Salas, who championed this effort. We're delighted to welcome Assembly Member Salas and to thank him for his leadership in the creation of this funding to the California State Budget Act of 2018-19. The Budget Act provides support not only to UC, but to the California Department of Public Health and the Valley Fever Institute at Kern Medical in Bakersfield. We're thrilled that they too will participate in today's webinar. They are important partners in this effort. Assemblymember Salas represents California's 32nd Assembly District located in the Central Valley. He's a UCLA graduate with degrees in history and political science. Assemblymember Salas is blazing a distinguished career in public service. He's worked in the White House as a legislative staffer in the state capitol and in the Central Valley as a district director, and was the first Latino city council member in Bakersfield. Since his election in 2012, Assemblymember Salas has focused on issues ranging from agriculture and water to workforce development to public safety and public health. He has been named Legislator of the Year by the California Police Chiefs Association, the American GI Forum of California, the California Issues Forum, the California Latino Capital Association Foundation, the Foundation for Independent Living Centers, and the California Optometric Association. Assemblymember Salas, thank you for joining us today.
Thank you. Thank you for having me this morning. Can you guys hear me okay? All right. Okay.
Yes.
Okay. Good morning. Thank you guys for having me. I'm really pleased that you guys have the Valley Fever webinar going on, connecting everybody up and down the entire state and across the country. You know, this work that you do is just so vitally important. I can't tell you how many times I go to community events up and down the entire state, and especially here in the Central Valley, where I hear somebody's story about how they've been affected by Valley fever. You know, a lot of times people are telling me stories about their grandparents, their parents, or one of their loved ones, a cousin, a niece, a nephew. You know, every time I'm talking about Valley fever, I'll ask the audience and I'll ask them how many people here know somebody affected by Valley fever, and you'll be surprised at the amount of hands that go up. That's why, you know, our efforts at the state level were so important. We wanted to make sure that we did something on Valley fever this year. In this year's budget, we were able to push for $8 million $3 million of that will be going to the University of California to help you guys with everything that you're doing to help advance the research on Valley fever. The other $3 million is going to go to develop the Valley Fever Institute at Kern Medical Center, and $2 million for the Department of Public Health, just because we know the sooner you catch this, the better the health outcomes are for all the families. What I can say is for the $3 million for the UC, I was reading some of the research that's being done throughout the entire system, and I'm very encouraged by what's being done and what you guys are doing. I did have a chance to visit Dr. Katrina Hoyer and Clarissa Noble over at UC Merced. I believe it was a year ago, and so got to see their labs. I've been up and down the entire state. I've seen some of the BSL-3 labs that have, I think we have almost 5 now, I believe, in the UC system, which is exciting. So I'm hoping that all the research, everything that you guys are doing up and down the entire state, connecting with each other, and then also connecting with the state of Arizona and what's being done nationally to help address this vital issue. I know every time I talk to Valley Fevers of America or to any of the families that are affected, they always ask two simple things. When are we going to have a more effective vaccine? When are we going to have a cure? Right? And I know that Nix-Z was discovered in the 1970s, and then didn't go to trials till many, many years later in the 2000s. But that's why what you guys are doing is so vitally important. And then understanding the different strains of Valley fever, how it affects each demographic up and down the entire state. Even my discussions with the Children's Hospital, they talk to me about the thousands and thousands of kids that they see every single year that they diagnose with Valley fever. So the work that you guys are doing is incredibly valuable. It's incredibly important. And I just want to thank you guys for everything that you're doing. And I'm more than happy to answer any questions. If not, I want to learn more about what you guys are doing, what's being done. And, I'm just excited for you guys. And I'm happy to play a little part in getting some of the funding up at the state level. I can tell you that every time I talk to my colleagues up and down the entire state, the first question was, what's Valley fever and why do you care? Why I care was very easy. It's just talking about the families that have been affected and relaying their stories and what they're doing. And I'll share with you guys just one story. I had a gentleman come talk to me, and he was a very proud man, a man that worked really hard. And he was saying, I went to work every single day. I got diagnosed. He was like, I wasn't going to let that deter me. So he continued to go to work, and he would work until he started falling over. And his supervisor said, you know what, I appreciate your heart and appreciate what you're doing. Yeah, we just can't have you falling over at work. And so, the work you're doing is vital. And I just want to say that I'm here to be supportive. Know that the Capitol and all my colleagues, everyone that supported this legislation and supported the budget allocation, is behind you. We were able to pass 3 out of 3 bills on Valley fever this year to help with the reporting numbers, to help with diagnosis and actually getting kits up and down the entire state for health professionals. So with that, I just want to say thank you. Thank you, Kathleen, Ms. Irwin, and thank you guys for having me this morning. And I'm more than happy to answer any questions. If anybody has any questions about exactly how this went about and how we actually got the money, I can share some of those stories. But if not, then I'll just say thank you guys for the work that you do, and just know that it's very impactful.
Thank you, Assemblymember Salas, for your remarks and for your support of the Valley fever research efforts here at the University of California. I'm Bart Aoki, Executive Director of the Research Grants Program Office at the University of California Office of the President. I'd like to take a few minutes prior to doing, are you still on, Assemblymember Salas?
I am.
Yeah, perhaps just a brief question. What are your thoughts? We really do appreciate your efforts on behalf of all of us in California in terms of Valley fever. what are the prospects for, or your plans for the next year in terms of any legislation at the state level in terms of continuation of these efforts in valley fever?
Well, we know this is going to be an issue for years to come. It has been an issue for years past already. We were very successful in the initial allocation, the $3 million directly to the UC, but I see this as an ongoing effort, and that's why we had legislation to help shore up the numbers. Things on the reporting guidelines, for instance, to show the need is there and that it's continuing to grow. You know, anecdotally, I could tell you up and down the valley and every community event I go to, everyone knows somebody that's been affected by valley fever. And so, you know, I hope that the legislation, once it's implemented and through the Department of Public Health and with what you're doing over at the UC, that we can show the need for why we need to continue to advocate for more additional funding in the future. So I see this as a good first step, but it's definitely not the last one, and we'll continue to go back to bat and try to get additional funding in the future as well.
Great. Thank you so much. Thank you for joining us also.
Thank you.
So I'd like to take a few minutes to briefly outline today's agenda and goals for the webinar and to mention a few housekeeping items. Over the course of the morning, we'll be hearing from the California Department of Public Health, who will provide an overview of cocci epidemiology and public health impact in California. We'll be listening to 3 brief research co-presentations focused on the pathogen and its environment, the host response, and clinical aspects of valley fever. We'll be learning about valley fever research and outreach efforts at the federal, state, and regional levels, and finally participating in a moderated discussion towards the end of the webinar. We hope that these discussions will provide an overview of the current landscape of valley fever research at UC, identify research gaps, and provide direction for promising future efforts. We'll keep each segment to the allotted time, and we want to allow as much time for discussion as possible. We'd like the webinar to be as interactive and informative as possible, so we've scheduled time for questions and discussions throughout. Although attendees are muted to reduce background noise, there are several ways for you to participate. Questions can be submitted at any time through the Q&A link at the bottom of your Zoom screen. And we'll pose the questions to the panelists during the panel sessions and during the moderated discussion. Kathleen Irwin and Uda Grishammer, our moderators, will direct as many of the questions as possible to our speakers, and they may combine or shorten some of them in order to do this. Additionally, we'll be seeking your input through the use of 2 poll questions, which we will launch on your screen during the moderated question and answer session at the end, towards the end of the webinar. We'd like to have you respond to these promptly, and as soon as you do, the poll will disappear from your screen. If your comments are not addressed during the meeting, or if you have additional questions or input, we'll also be sending a post-webinar survey to everyone who registered to the email address you entered with your registration. Look for that email in the next 48 hours, and please provide us with any additional feedback or ideas. Your feedback will be used to inform the development and structure of an RFP, which we expect to issue by the first week of December. Again, we thank you, each of you, for taking the time to join us today. I'm now pleased to introduce our next speakers, Dr. Seema Jain and Ms. Gail Cooksey from the California Department of Public Health Infectious Disease Branch. Dr. Jain is the chief of the Disease Investigations Section. Her work focuses on surveillance and outbreak investigations for many infectious diseases, including the coccidioidomycosis. Ms. Cooksey is the mycotic diseases epidemiologist. Her work focuses primarily on coccidioidomycosis surveillance, outbreak investigation, and outreach. They're joining us to provide an overview of cocci epidemiology and its public health impact in California. I'd like to welcome Dr. Jain and Ms. Cooksey.
Good morning, and thank you for having us and giving us a few moments today to present the public health perspective on coccidioidomycosis. Public health is sometimes overlooked amongst the many clinical, diagnostic, and treatment challenges of cocci, but there are some very important public health questions that need to be answered, and these answers could have a really big impact on disease incidence statewide. Next slide, please. Coccidioidomycosis is a reportable disease in California, and both clinicians and laboratories are required to report all new cases of cocci to the health department. Since 1995, CDPH has received individual reports of COXI cases and has used these data to track trends. This figure shows the number of COXI cases in the blue bars and the rates of reported cases per 100,000 population in the black line. In 2017, nearly 7,500 new cases of COXI were reported for a rate of 18.8 per 100,000 population. And this is the highest annual incidence of reported cases since COXI became reportable. This was a 213% increase compared to 2014. In addition to case surveillance, CDPH also reviews hospital and death data and has noted increases in those trends as well. In general, over 1,000 individual patients are hospitalized with COXI in California each year, and nearly 80 California residents die with COXI as a cause of death. With these increasing trends and the potential for severe disease with COXI, one of the most pressing public health questions that we face is what's causing these recent increases in incidence. Is it increased diagnosis or reporting, increased construction activity or dirt disturbance, the movement of susceptible persons to high-risk areas, or changes in climate or environment? And without answers to these questions, we are limited as public health in our ability to provide guidance on how to prevent further disease. Next slide. In addition to these individual case reports, CDPH also investigates COXI outbreaks. These outbreaks are largely occupational and associated with jobs that involve dirt disturbance. Previous outbreaks have occurred involving different types of construction work, wild and firefighting, archaeological excavations, and even a site where a TV show was being filmed and dirt was disturbed for a music festival scene. Several such outbreaks are shown on the map on the left of this slide. And the map also indicates the county level rates of COXI, with the darker blue color indicating higher rates. As you may note on the map, some of these outbreaks have occurred outside of the counties where the highest rates of COXI are typically occurring. It's challenging for us as the health department to provide guidance on which potential work sites may present a risk for COXI. Our case data is based on patient residence and will not reflect where a person was actually exposed to the fungus or how risky a particular plot of land might be. It's a major challenge for us to provide appropriate guidance on when and where the workers and the surrounding communities to a work site might need to take extra steps to protect themselves from COXI. Often the workers involved in occupational outbreaks are exposed to substantial amounts of dust, and previously healthy young workers can become severely ill, sometimes with disseminated disease. Their symptoms, particularly severe fatigue, can persist for months, preventing these workers from returning to their jobs. We've investigated more than 6 potential outbreaks in the past few years, and the largest outbreak was among solar farm construction workers and involved 44 confirmed cases of COXI, 9 of which were hospitalized. 34 of these workers missed work for a median of 22 days, and 2 ended up getting diagnosed with disseminated disease. And a survey of those people who worked on this site where the outbreak occurred identified another 89 potential people who had suspect COXI, which just highlights the fact that there may be a large number of undiagnosed cases from this outbreak. Our occupational health colleagues at the health department are often asked by local health departments, planning departments, safety officers, unions, and employers about what can be done to reliably protect workers from COXI infection. Is it respiratory protection, dust control measures? Our occupational health branch has developed detailed guidance available on our webpage, but research is needed on how effective these different recommendations actually are, and new ideas and tools for prevention are very much needed. The health department's limited in our own resources to investigate these questions, and we strongly encourage UC researchers to consider major public health questions when planning their research. Thank you for allowing us to speak, and please feel free to reach out to us anytime with questions. Thank you.
Thank you, Ms. Cooksey and Dr. Jain. Your overview demonstrates the significance and public health impact of valley fever in California and really helps highlight the importance of research, and hopefully you see research in addressing some of these issues. It's very informative. I am Kathleen Irwin. I'm joined here by Ute Grieshammer, and we are going to now turn to 3 research presentations in the area of valley fever research that Bart Aoki already introduced. In each of the upcoming segments or co-presentations, we will hear from 2 University of California experts, followed by a moderated question and answer session. As a reminder to the attendees watching the webinar, please feel free to submit questions throughout the presentation by using the Q&A icon at the bottom of your screen. And Uta and I will do our best to then moderate a discussion after the presentation. So I turn now to our first co-presenters, and I am delighted to introduce to you two speakers on the topic of the pathogen and its environment. Dr. John Taylor is a professor in the Department of Plant and Microbial Biology at the University of California, Berkeley. He has studied the evolution and ecology of the cocci species for more than 25 years and is currently working on detection of this fungus in the environment and on understanding how genetic variation in the fungus relates to variation in its pathogenicity. Dr. Taylor is going to be joined by Dr. Anita Sill, who is a professor in the Department of Microbiology and Immunology at the University of California, San Francisco, where she runs a biosafety lab, level 3, BSL-3 lab facility to study fungal pathogens that infect healthy humans. Her current research includes the molecular analysis of the coccidioides fungus. Welcome to Dr. Taylor and Dr. Sill.
Thank you very much. If I might have my next slide. So I'm going to address 4 questions today. Where is the disease, coccidioidomycosis? What's the life cycle of the fungus that causes it, coccidioides? Why is coccidioides in hot, dry areas of California? And finally, do I need protection when I work outdoors? Next slide, please. So as Gail and Seema said, coccidioides is in the environment. It's only in the New World. It's in North America, Central America, and Latin America in hot, dry areas. So you can see it's in the desert Southwest in North America. It's on the Pacific side of, say, Guatemala in Central America, and in the rain shadow of the Andes in Latin America. Next slide, please. And Gail already showed a map of California, and I've got one too because I want to emphasize it's in the southern San Joaquin Valley. The incidence of the disease is a function of the local population and the presence of the fungus. So you can see Kern County and Fresno County, which have the cities of Fresno and Bakersfield, have a lot of cases, but it's also in coastal California, San Luis Obispo County, where the solar farms are being put in. And then in Northern California, it's in the San Francisco Bay Area in eastern Contra Costa and Alameda counties. And it's also further north. It's been found in Yolo and Glenn counties. Next slide, please. So the circle or the ellipse down there shows the area where the most coccidioides is present in the environment. And that is about Assembly District 32. So we all owe Rudy Salas a vote of thanks for bringing the importance of this fungus to the attention of the California State Legislature. Next slide, please. I want to talk about how coccidioides undergoes its life cycle. Outside of an animal, it grows as a filament outside of a living animal. And that filament breaks up into little spores, and you can see those little spores. They're called arthroconidia. Next slide, please. And they spread a short distance in the soil or a long distance in the air. Next slide. And they're inhaled by small mammals in nature. And inside the small mammal, that arthroconidium, next slide, please, converts to a spherule. Next slide. And you can see that this spherule, next slide, is about 1,000 times bigger than that arthroconidium. And it produces spores inside of the spherule, which can be released if the spherule ruptures. And these spread the fungus throughout the small mammal. Next slide. However, most of the time, more than 90% of the time, the small mammal wins this battle, and it walls off that enlarging conidium in a prison of its own immune response cells. In that case, the small mammal has a long life because it doesn't develop disease. If the spherule ruptures, it has a short life because of what's called disseminated disease. Next slide. In either case, the small mammal has a relatively short life by human standards, And when it dies, the fungus, which is alive in that granuloma or alive after dissemination, now converts back to the filamentous form, digests the animal protein, and produces a tremendous number of arthrokinetia. Next slide, please, which then continue the life cycle. Now you'll notice I haven't mentioned humans in this life cycle. Next slide, please. And that's because we were not in the New World when this disease evolved. And we've come in the past maybe 20,000 years at the most. But we behave like the small mammal. We inhale the spores, and then two things can happen. Most of the time, more than 90% of the time, we wall it off in the granuloma. And occasionally, and a small percentage of the time, we get disseminated disease, which unless treated is fatal. Next slide, please. So there are models on the presence of coccidioides in the environment, and almost all these models now look at climate and soil as affecting the fungus. Next slide, please. But we now know that coccidioides lives in these animals. In fact, it's eating these small animals. So the models need to include the animal. Next slide, please. And then those animals are herbivores. They eat plants. So we need to include plants. Next slide, please. Next slide. And so this, I think, is the better model for the presence of coccidioides. Next slide, please. And I think this route from climate to plant to animal to the presence of coccidioides explains why changes in the climate are reflected in changes in coccidioidomycosis, the human disease, the year following the change in the climate. Next slide, please. Now, why is cocci in the hot, dry areas? In a hot, dry area, one of the best sources of water and food is a small mammal, 70% water, a lot of animal protein. And so cocci, by living in that small animal and not causing disease, is poised to be the first microbe, the first fungus to gain access to that food resource when the small mammal dies. In areas where there's more water in the environment, it's not such an advantage, and cocci is not such a good competitor. Next slide, please. So now the final question, and this is one that Gail and Seema addressed it, do I need protection? So one can sample the soil to see if cocci is present, and you'll notice this is a small mammal burrow. This sort of sampling has been done since the 1940s. I've done it as well as have researchers in Baja California and Arizona, and we can find Coccidioides immitis or Coccidioides species in the soil, especially in mammal burrows. Next slide. However, mammal burrows soil sampling is labor intensive and expensive, and it would be better if we could sample the air to see if Coccidioides is present. And so I've set up 15 air samplers in the western edge of Kern and Kings and Fresno County, where Assemblyman Salas knows very well, to see if I can detect this in the air. We have almost a year's worth of samples now, but we haven't analyzed them. We're going to do that shortly. And then next slide, please. The other way to monitor would be look at the small mammals directly. And this was done in the 1940s. It's being done now in Baja California, but it's not been attempted in California. And again, it would be labor intensive and a relatively expensive way, but a sure way to know about the presence of coccidioides in that environment. Next slide, please. So the sampling angle is the same thing that Gail mentioned. There are two ways to deal with coccidioidomycosis. One is to treat the disease, and the other is to prevent it. And so we're working on the prevention side, and in fact, we've worked with the California State Department of Health by providing soil samples that have coccidioides, and they're able to isolate the fungus from those samples for further research. And that brings me to my colleague Anita Sill, who's going to do the next part of this presentation on the molecular aspects of virulence in this fungus. Thank you very much.
Thank you, John. I hope everyone can hear me. I'm just going to remind people that I'm a molecular geneticist who studies fungal pathogens. I've been studying a different soil fungus related to coccidioides for the past 19 years, and over the past year and a half, largely due to inspiration from John, I've been applying the skills and lessons that I've learned from studying that pathogen to studying coccidioides. and based on my experience, I want to make the argument today that one very important focus for coccidioides research is thinking about the basic biology of the fungus itself. Assembly member Salas already mentioned that vaccines and other targeted antifungal therapeutics are really critical tools to fighting coccidioides infection. And I can't emphasize enough how important it is to understand more about the molecular biology of this organism in order to develop those tools. So I'm going to talk about how basically, identifying some of the critical questions in this organism. So let's go to the next slide, please. So John already mentioned that there's a dramatic transition that coccidioides undergoes between the time when it's in the soil and when it's found in a mammalian host. That these arthroconidia, these spores, get inhaled and transition into this, undergoing this amazing developmental program to make spherules that each contain endospores that when the spherule ruptures, those endospores can disseminate and cause disease. And I just want to say very little is really understood about the establishment and the maintenance of this developmental program. How does the organism switch from the soil form to the host form? And really from that point on in the host, we still understand very little about the coccidioides factors, the fungal factors that are critical for manipulating the host immune response. And we're going to hear some more about the host response a little bit later. Next slide, please. So based on what we know about all pathogens and sort of decades of research on thinking about infectious disease, I think these are some of the critical questions for coccidioides. So first, which fungal molecules are important for the establishment and maintenance of the host form? How does the fungus, once these spore particles are inhaled, how does the fungus make this spherular structure full of endospores? What are the factors that regulate that process and determine the pathogenic form of the organism? And then which factors are important for the ability of the fungus to escape the innate immune response? What are the fungal factors that manipulate phagocytic cells, such as macrophages, which are designed to take up foreign invaders and kill them? How does this fungus escape? And perhaps those lines of research would lead us to tools that might allow us to augment the host response to the pathogen in susceptible individuals. Next slide, please. Okay, so how do I recommend that we go about answering these important questions? Well, I want to make the point that this is really a propitious, amazing time to apply molecular approaches to coccidioides biology. This is really an understudied organism, and there are, let's say, 9,000 genes in the coccidioides genome, and we know a function really of a very, very, very tiny fraction of those genes. How do we sort through those genes and identify the ones that are really key to the ability of this fungus to cause disease? And I think one of the answers, and the reason it's a propitious time, is because it's really, we are really benefited by modern molecular biology to be able to take these approaches. So I think the answer is really genetics and genomics. And our ability to do these approaches is fueled by, really recent advances in, the ease of genome sequencing and as well as genome editing. So for example, GWAS, Genome-Wide Association Studies. These are studies John and I, in collaboration with Rachel Brem and colleagues at UC Merced and colleagues at UC San Diego and UC Riverside, and you'll hear from some of these folks today. We're taking this approach where the idea is that we are querying different coccidioides strains, and we're looking at the molecular basis of variants in those strains. For example, we can phenotype those strains in the laboratory. We can look at whether there are particular strains that are better at making infectious spores or better at doing that transition into the host form. And then we can identify the actual changes in the genome that allow those strains to be better at those processes. And that will lead us to fungal factors that are important for disease. And this is really something that can be done now because of the ease of sequencing genomes. Forward genetics has been a really powerful approach in many systems for letting the organism tell you which genes are important, looking, for example, for mutants that can't make the transition, or reverse genetics, taking advantage of genome editing technologies, looking for identifying genes that are implicated in various processes by virtue of their function in other organisms, and then mutating them in coccidioides and understanding their function in disease progression. I think that these approaches are incredibly powerful. There have been advances in molecular and cell biology that allow us to do things like look at all the genes that are expressed in the soil form versus the host form and identify factors that promote the biology of the host form. Similarly, proteomics technologies have advanced. It's possible to probe key regulatory circuits by a variety of molecular techniques to identify the targets of regulatory circuits that drive establishment and maintenance of the host form. And advances in cell biology, imaging, thinking about the host response, doing many of these studies in vitro and then validating them in vivo. It's really an extraordinary time to delve deep into coccidioides biology. And the potential impact of these approaches, as I mentioned, is identifying fungal targets that could have the potential to be vaccine candidates, or could even be the basis of developing coccidioides-specific therapeutics. So I'll finish there and open it up.
Drs. Taylor and Dr. Sill for your presentations. Those are very informative and helpful. And we wanted now to open up the next few minutes for some question and answer. So we're glad you're both back on screen. We have some questions that have been coming in from our attendees, and we hope that those of you listening, if you have additional questions, please feel free to submit them.
So I'll start with the first question, there were two questions that were actually related, and John already answered one regarding how air sampling is performed and the sequencing technology that you use to analyze those results. But maybe you could also elaborate a little on whether there have been previous efforts to isolate the fungus from air samples and what the outcomes of those studies were.
Sure. So the air sampling that I've been doing is a very simple air sampling because the samplers need to be inexpensive if you're going to deploy them over large areas of the Central Valley. And so what we use is we use an empty dish with no growth medium in it, and we just leave it out and let the spores settle in the dish. And we developed this for studies of indoor air, and it works quite well. And then you simply swab out the fungi, you also get the bacteria that have settled into the dish, isolate the DNA, and from the DNA you can identify which fungi and which bacteria are present. And there's a particularly sensitive test that's been developed in Arizona by Bridget Barker at Northern Arizona University. There's also a very sensitive test developed by the California State Department of Health. And so you can send the DNA to either of these groups and they can tell you if they can detect coccidioides. And so we've used that for soil. We don't have analysis from the air sampling yet. I'm hoping it's going to work because it'd be much less expensive and much less labor intensive than soil sampling. Could cover larger areas. But when I have, as I said, 11 months worth of samples, but I'm waiting to get the 12th month to send them off for the analysis.
Thank you. So, Dr. Sill, you mentioned sort of some of your enthusiasm and the excitement around genome sequencing and the ability to do some genetic editing. I'm wondering if you and if Dr. Taylor also wants to add to this, could say a few words about what you think are the potential breakthroughs that could happen in this area of research, let's say in the next 3 years versus, let's say, 5 to 10 year research agenda.
Yeah,
short-term, long-term kind of view. Yeah.
Sure. So short-term, so There are so many examples because we really understand very little about the molecular biology of this fungus. So short term, I would say the most compelling question is thinking about establishment and maintenance of the host form of this organism. So I didn't mention that one thing that's very helpful for coccidioides research is that you can recapitulate this transition from the soil form to the host form in the laboratory, which allows one to really study this in detail, and then validate data in an animal model to make sure that the in vitro studies parallel what's happening in vivo. So I think one of the key short-term questions would be looking at regulatory circuits that are required for that transition. And I think that that's a short-term goal because we already know some of the, based on transcriptional profiling, we already know some of the candidates, the transcription factors, which are the molecules that drive different developmental programs. We know what some of those transcription factors are that show enhanced expression in the host form, and we have the tools there to query their function in that transition. So I think that would be a short-term goal. I think a longer-term goal would be this, or deliverable would be this, a deeper understanding of the ability of this fungus to manipulate the immune response. It's obviously able to manipulate the innate response. It persists in hosts for many years. I think there are some compelling and exciting longer-term experiments comparing the ability of different coccidioides strains like natural variants to compete in the host environment and establish that long-term niche. Those would be amazing experiments because they might allow us to identify pathways in the fungus or even in the host that influence that long-term persistence, which could then have implications for shortening the extent of disease progression in people.
Yeah, I would agree with that. I think basic research about how the fungus lives in the host is really important because the commercial concerns that develop drugs or develop vaccines can't do that kind of research. They don't have the funds for that. The other thing is anything that relates to prevention, because drug companies, vaccine is prevention, but drug companies, prevention isn't what they do. They treat the disease after it's been acquired. So the role of the university, in addition to basic research on how the fungus behaves, is basic research on where is the fungus and how to prevent acquisition of the disease.
Thank you. So along the lines of using modern tools or state-of-the-art tools of analyzing the fungus itself, an attendee is curious whether you could elaborate on what types of systematic proteomic studies have been performed to understand the life cycle of the fungus and its ability to manipulate the immune response.
There's a lot to be done in terms of proteomics to do a really detailed and comprehensive study on a proteomics level of the transition between this spore form and the host form. That has essentially not been done in a very sort of modern and rigorous way. Similarly, looking at the interaction of this organism with host cells, there are some published studies on, say, for example, macrophage interactions, but there is a lot yet to do on the molecular level, not limited to proteomics, transcriptomics, as well as proteomics. One could really analyze, these days when people do infection models and proteomics analysis of host cells, they're not even just limiting themselves to understanding which proteins are upregulated or downregulated in either the host cell or the pathogen in response to infection, but also looking at other more subtle but key regulatory changes in the host cell, such as ubiquitination of host proteins, reflecting the ability of the pathogen to manipulate stability of different host factors. This has not been looked at all for coccidioides.
Another aspect of that is that There is a lot of literature on fungi that live inside plants without causing disease until the plant dies. They're called endophytes. Cocci may be the model endozoan, a fungus that lives inside an animal without causing apparent disease until the animal dies. And it's possible that there are many other microbes in our bodies that are endozoans and evolved in wild animals or in humans before civilization came around. And so lessons learned from Coccidioides and how it manages to exist in the animal without causing disease, as well as causing disease, could be very important.
Thank you. So we have a couple questions regarding the sequencing of the fungus. One relates to, I'm going to just combine them and you can address them together. One of them relates to some sequencing of Coccidioides that was done as part of the Valley Fever vaccine project about 10 years ago, and some sequencing that was done at that time, and whether or not that sequencing is being used now by microbiologists. And then what about current sequencing methods and the time it takes and the process for doing that?
So in the 2000s, we began to understand the species of Coccidioides. There's one in California, Coccidioides immitis, and then the one pretty much outside California, Coccidioides posadacei. And there were sequences, there were also understood that there were populations that had genetic differentiation within each of those species. That information allowed NIH to fund sequencing of one individual from each of those populations, or actually two individuals from each population. Those sequences were done with the old Sanger expensive sequencing approach. And those are available, I believe they're available at FungiDB, Fungi database. Since then, sequencing has gotten less expensive, and so Bridget Barker at Northern Arizona has sequenced literally hundreds of isolates, mainly from Arizona, but also some from California. And as part of the project that Anita mentioned that we are in along with UC Merced, UC Riverside, UC San Diego, we have sequenced, I think, 30 genomes of Coccidioides. These are much less expensive to sequence now. And for the environmental sequencing, you could combine several hundred environmental samples to do sequencing of regions of DNA that you'd amplified from those samples in one sequencing run, relatively inexpensive. So sequencing is definitely less expensive, and the original NIH sequences are publicly available, and as are many of the more recently sequenced genomes.
But I'd like to, so thank you, Don, that was an amazing summary. I just want to emphasize that one of the things that we're trying to go beyond just the genome sequence. So one key thing is the phenotyping that we're doing to basically allow us to utilize that genome sequence to identify genes that are important for basic aspects of the fungal biology. And one really important thing is that once you have a set of strains where you can do GWAS analysis, you have those genome sequences, and then down the road you can phenotype in any way that becomes of interest and use those existing genomes to identify fungal variants, the SNPs that are associated with different traits. So it's a very powerful tool for the whole community because any group can do the phenotyping that they're interested in and identify important traits in this fungus based on the sequence that will be provided to the community.
Well, we want to thank you both very much for your remarks and also for addressing the questions. We did have a few other questions that have come in, and as you know, we're having a moderated discussion, so we can bring some of those back toward the end of the webinar. We're going to turn now to our next panel.
Yes, so we will now look at the response of the host to cocci infection. And so in the second presentation, we will hear from Dr. Joshua Farrer and Dr. Katrina Hoyer. Dr. Fair is the Marcy and Michael Oxman Professor of Infectious Diseases at University of California in San Diego and Chief of Infectious Diseases and Director of Microbiology at the VA in San Diego. His research focuses on host-pathogen relationships using animal models of infection, and he has been interested in valley fever since moving to California in 1969. Dr. Hoyer, who will join him a little later, is assistant professor in the School of Natural Sciences at UC Merced. Her research interests include to better understand what constitutes a productive immune response in patients who properly control cocci infection as compared to the immune response of individuals who develop disseminated disease. I just want to remind everybody the attendees to keep asking questions using the Q&A box, and now I'll turn it over to Dr. Fair.
Thank you very much. It's a pleasure to be here. First slide. So this talk is going to be about 1,000-foot-high examination of the immune response. And the first point that I want to make is that the vast majority of these infections are self-limited. Some of them are symptomatic, producing pneumonia of various severities. Others are quite mild and probably don't come to medical attention. And we believe that a large number are completely asymptomatic. But regardless of the severity of the infection, second infections are extremely rare. So this means that most people are able not only to make a protective immune response that controls the infection, but that becomes an adaptive immune response that generates memory T cells and protects people from further infections, even though they continue to live in the endemic area. Now, not everybody, however, recovers. And I'm not going to talk about people who are immunocompromised, except as that informs us about the necessary immune functions. But about 5 to 10% of infections spread outside the lung, becoming disseminated. And by dissemination, we mean that infection outside the lung is clinically apparent. Dissemination probably occurs in everybody as part of the primary infection, which is similar to tuberculosis. But clinical dissemination can occur immediately, that is while the pneumonia is present, or only becomes apparent months to years after the original infection. Now, this dichotomous response to the infection is not completely understood. So on a broad level, we know that people who recover make very low titers of antibody to fungal antigens, which are mostly secreted proteins, but they do develop delay-type hypersensitivity, which is measured as a skin test reaction analogous to the PPD in tuberculosis. Whereas in contrast to that, people who disseminate make high titers of antibody and they don't develop DTH. If you do develop DTH, it lasts about 10 years if you leave the endemic area. So that's further evidence that you have generated memory. The reason that some people make one response and other people make another response is really not understood. But we do know that the dissemination indicating an inadequate or inappropriate immune response to the infection is much more likely to occur in African Americans and Filipinos, 3 to 10 times more likely to occur than in European Americans. And this is an amazing difference. Next slide. So what consists of an adequate immune response? Well, we know that the development of an interferon gamma pathway is absolutely required because people who are born with inactivating Mendelian mutations in this pathway develop disseminated cocci when they're exposed to the infection. We also know that CD4 T cells are required, and that comes from the AIDS epidemic, where there was an inverse correlation between your risk of dissemination and the number of CD4 T cells that you had when you got infected. We also know that TNF plays a role in immunity, and that's because people who are treated with immunotherapy, anti-TNF therapy for other conditions are more likely to develop disseminated disease. This isn't as profound as having no CD4 T cells, but it is a measurable risk factor for dissemination. But I want to emphasize that in the vast majority of cases, the susceptibility to this infection is not due to inactivating mutations in these genes that play a central role in immune responses. As those mutations make people susceptible to many infections, many opportunistic infections, whereas the people who disseminate, who have disseminated cocci, have really they're healthy people, previously healthy people who have no history of opportunistic infections and subsequently do not develop opportunistic infections. So this is a very subtle genetic defect, if you will, that that probably is unique to this fungus. Next slide. So what do we not know? Well, we don't know a tremendous amount. We know in general that the innate immune response guides the adaptive immune response. And we don't know whether people who disseminate make a different innate immune response to the fungus than people who do not. That's certainly true in mice, but we don't know whether it's true in people. We don't know which genetic polymorphisms determine the kind of immune response that people are going to make. Everybody makes an immune response, but some immune responses are good and some are not. And we don't know if there are important differences in organ-specific immunity to the fungus. For instance, it's not uncommon for the pneumonia to have completely resolved and a joint or a bone infection to progress inexorably. So clearly there are differences there. We don't know if the fungus secretes enzymes or secondary metabolites that could subvert the innate immune response, for instance, interact with the aryl hydrocarbon receptor and so generate T suppressor cells. And we really have very little idea about the composition of the spheral and endospore cell walls. And this is how the fungus interacts directly with cells. So we don't know all of the carbohydrates and their structures that are present on the fungus and how they would interact with C-type lectin receptors and other pattern recognition receptors. The exception to that is the beta glucan interaction with dectin-1, which has been well studied in mice. However, inactivating mutations that occur in that receptor in people have so far not been associated with risk of disease. though we don't know how many people in the endemic area have that mutation. So there's a lot to be learned. Next slide. And now I'm going to turn it over to Professor Hoyer.
Thank you, Josh. So Josh has given you really the big, broad overview on what we know and what we don't know. I'm an immunologist primarily focused on adaptive immune responses thus far in my career. So I'm going to start there and go into a little bit more detail. As Josh told you, we know that antibodies are made. We can measure this through the skin test and through other clinical assays. But the role has been less clear of what antibodies are doing in the disease. And there's been suggestions that it's important, but also that it is kind of opposing a productive immune response. We also know that T effector cells are critical. We know particularly from most of the human studies thus far that Th1 cells are important for fungal clearance. But we know primarily from other fungal diseases that Th17s are absolutely critical for the ability to clear other fungal species. In particular, Paracoccidioides, a similar fungus, Th17s are absolutely required. And we know a little less about the Th17s in this particular fungal disease. There have been a couple of studies indicating a role for CD8 T cells. Josh's early work suggesting that CD8s do have an important role in clearing the fungus. But then there are counter publications indicating that there is no role for the CD8s. So you can see already that we know very little about the adaptive immune response. Regulatory T cells, which are really big in kind of the autoimmune and cancer field, we know very, very little in this particular disease about the role of regulatory T cells. There have been a couple of association studies between regulatory T cell elevation and the inability to clear infections in mice. We also have a recent pediatric study suggesting that the increase in regulatory T cell frequency is associated with an inability to clear this fungal infection and results, in chronic infection. So overall, clearly I'm giving you a lot of things that we don't know. And so I think it's really important to understand, as Anita said, and Josh, what's happening at the adaptive and innate immune response and understanding how they are interacting with each other to produce a productive or adequate response for this fungus, and also the mechanisms of this process. So next slide. So adaptive immune cells and innate immune cells both mediate many of their functions via cytokines. One of the ones that we know the most about, in addition to TNF that we heard about earlier, is IL-10. We know that in IL-2 deficiency, particularly in mouse studies, that there is a higher fungal burden and that these mice are unable to clear the infection. But we know that IL-10 is produced by both the innate and the adaptive immune system, including these regulatory T cells. So which cells are important for producing this IL-10 that is resulting in prolonged infection? We don't know. We know from several human studies that impaired IL-12 and interferon gamma signaling is associated with severe disease. We also know that STAT1 gain-of-function mutations are linked to dissemination, and this results in an impaired responsiveness to interferon gamma. But if we look at the interferon gamma signaling pathway, signals through interferon gamma that result in STAT1 gain-of-function also inhibits several other cytokine signals through STAT3, including IL-17. So a STAT1 gain-of-function mutation is also likely to inhibit IL-17 responses and those Th17 cells that I mentioned earlier. So we don't know a lot about how these mutations and gain-of-functions and signaling interactions are playing a role in the disease process. So which cytokines are critical effectors for fungal clearance are largely unknown. Which cells are producing these cytokines and responding to these cytokines during a coccidioides infection has not really been explored. And then really, how do these cytokines mediate immune responses and clearance of the fungus? And are there ways that we can manipulate these responses, as we've done in autoimmune disease and cancer, could have a big impact. So next slide. And as Josh mentioned, we really don't know much about the local tissue environments. And really this infection begins in the lung. You inhale the arthrochonidia and the transition begins where they change their life cycle into the spheroidal form. And so these local tissue interactions are likely shaping that early immune response, both at the innate and the adaptive immune level. And we really haven't explored what's happening there. We know from a lot of asthma studies that the lung microenvironment is inherently anti-inflammatory, that there are high basal levels of IL-10 secretion from the local environment. And many of the studies, as Anita mentioned, are done with bone marrow-derived macrophage and macrophage cell lines. And while we've learned a lot initially about the innate immune response, we really don't know much about how this fungus is interacting with the local alveolar macrophages, which are quite different from the bone marrow-derived macrophages. And then Josh mentioned, and so did John, these granulomas that form that are really important for walling off the fungus during infection. And really as immunologists, and even as biologists, we really don't know much about how the granuloma forms. We don't understand the biology of what keeps it intact. And really as immunologists, we largely know nothing about the granuloma formation, even though it is critical in dealing with this fungus. So I think we really need to understand the immune response at that first site of infection in the lung tissue to really understand how the host response develops to coccidioides. Next slide, please. So how does some of this translate to human studies and what is really lacking in what we know about human disease? So it would really be useful to discover correlations between the immune response and disease outcome that can be measured not only in the blood, where much of the work is being done, but also from lung tissue samples and other infected tissues such as the skin. It also would be useful to look at immune genetics and be able to compare the limited pulmonary and disseminated infection and be able to compare what's happening genetically in those two types of diseases. We need lots of immune functional data that describes what is a productive or adequate immune response that results in resolution of disease. And really, we don't have any longitudinal immune profiling. My group has looked at acute disease. Many others have looked at later stages disease, but we really don't have anything that compares and combines these two stages. And then what I would really love to do, which I think is probably the most challenging question here, is, again, we've talked about how 60% or more of patients have a very mild disease that resolves spontaneously. And clinicians don't typically see these patients during their infection. So what does this immune response look like? And if we could understand that, that would really give us, I think, a tool to being able to really understand how to develop a productive response. Now, how we ask that question, I think, is a lot more challenging, but I think that's really would give us a clearest understanding. And so I think we need to go back and forth between combining animal models and really getting at the mechanistic questions where we can eliminate genes and transfer cells and really understand kind of the basic biology, and then translate this over to the clinical samples to verify that these things are relevant to the human disease. And so that's where I will end. Thank you.
Thank you very much, Dr. Hoyer and Dr. Fierer, for your very interesting remarks and insights into the adaptive and innate immune response to cocci and the differences between the controlled and disseminated disease. And it really sounds like understanding that difference is critical, would be critical in helping develop prevention and treatment. Also enjoyed your thoughts on the major knowledge gaps and the research that's needed to advance treating and preventing human disease. So we'll now go into the first question.
Well, our first question, I think, is going to be similar to one we asked the last two panelists in this really exciting range of topics that could be pursued in research. What do you think would be achievable and really an important focus point, perhaps, in a short-term 3-year time frame as compared to sort of a longer-term 10-year research agenda? Of course, all of it could go long-term, but in terms of really seeing some important breakthroughs.
So I think that one advantage of having a UC-wide initiative is that it will greatly increase the participation of investigators in this problem and access to patients with this infection. And we really need to know whether what we find in inbred mice, which is a very easy system to manipulate, bears on the human response. And I think it's only by studying humans who have this infection that we're going to learn that. And I think we can do that with the cooperation of all the UC medical centers. And I think it's possible to get a lot of information in the next 3 years.
I would echo that that Josh has just said. I think we have the strength in the UC system and the technology to be able to really make some major steps forward in both acute disease and understanding what is happening there. If we can get a hold of things like skin biopsies and lung samples, in addition to the peripheral blood samples that are much easier to get, to be able to really look at not only the genetic components, but also the immediate host responses. And then to be able to take what we find from that and be able to go back into mouse studies and manipulate in that more kind of manipulable system to ask if how these things are working mechanistically. And I think that could really get done in the short term. I think we could make some good steps forwards that would then give us a lot more questions to ask for the longer term.
And I want to add one other thing, that is the immunogenetics involved here. And having a lot of cooperation among all the medical centers would again allow the collection of much more DNA from people who have this dichotomous immune responses. My colleague George Thompson at UC Davis on his own collected a few hundred DNA from a few hundred people, but that's a relatively small sample size for doing these kinds of studies. And I think having resources to get more samples will be tremendously important.
Thank you. We have a question from an attendee who's curious, and just to be clear, in what context can cocci behave as an intracellular pathogen? Can you comment on that?
Yeah, so it's a mixture. The spherules are enormous when they're fully grown, and the only cells that they're ever in are multinucleated giant cells. And but however, the endospores are quite small and are clearly ingested by phagocytic cells, and as they enlarge, they're small spherules, and they're also taken in by cells. So it's different from the yeast infections like histoplasmosis that Anita studies, in that organism spends all its time inside macrophages pretty much. But cocci is both intracellular and extracellular.
So did you want to add anything, Dr. Hoyer?
No.
No, okay. So you did talk really in your remarks about the importance of the sort of translation between the mice studies and humans, and how that can really potentially lead to better interventions, either prevention or treatment in humans. At this point in the knowledge that we have and that you have, are there particularly promising targets that you think would be worth pursuing in the host response pathways, or do you think it's still a kind of open question?
So Josh might have a different answer than me, but I feel like we've got some good ideas, but I don't know that we have enough evidence yet to move forward with targeting some of these ideas. So I started out as an autoimmune disease immunologist. So I think about this in terms of, using targets and using, cytokine and immune modulators that already exist potentially to be able to manipulate the immune response in coccidioides infection. But the problem is I think we still don't really understand what that productive response looks like. And so I think at this point, maybe some of the clinicians have a different perspective, but I feel like at this point we might be acting a little blindly. I think we need more understanding of what that early immune response looks like in association with disease outcomes to then be able to manipulate it. But I think there's a lot of evidence out there that we could be successful with manipulating the immune response with the cancer immunotherapies that exist and the autoimmune disease therapies that exist. So there are a lot of, I think, potentials out there once we have a better understanding. You have a different
Dr. Fira, did you want to add anything?
No, I think that was fine. We really, I think, have learned a lot from the mice in terms of vaccines, what vaccines have to be able to do to protect. But obviously we don't know how that translates to the human immune response.
I'd like to go back to your earlier comments in terms of leveraging the research capacity within the UC system, and also the amazing UC hospital capacity, of the 5 major medical centers, and the information can be gathered there. And Dr. Hoyer, you had mentioned how we really need to understand what the immune response is like in people who don't even present with symptoms. Do you see an opportunity in this, in this large database, that might exist in the UC system to identify the people? I mean, I'm sure that's the biggest hurdle, really, in trying to understand that part.
Well, yes. I mean, we have a lot of new people moving into the endemic region, and I think this is an opportunity to ask just that question. What does that productive, kind of mild cocci response look like? And I think the way to do this would be a little bit challenging, but I think you have to catch people as they move into the region and monitor them until they develop infection. I don't know how else to do that. If somebody has an idea, I'd love to hear it. But we've got. I moved to the region 7 years ago, and I'd love to find out when I get infected, because most of us don't realize when we're infected. And if you talk to people who live in the endemic region, most of them probably have antibodies, so they've been exposed at some point.
So we do have the advantage now that the skin test reagent is commercially available after about 2 decades when we didn't have it, and this is an excellent way of finding people who had asymptomatic infections. In fact, the skin test is the basis of our claim that there are asymptomatic infections.
I'd really like to catch them, though, at that acute phase of disease when they kind of are still in the infected stage, before they've had it, respond 20 years ago.
Well, here's a real blue sky question for you then. You mentioned in your remarks about one of the real mysteries is the folks who are able to launch an effective immune response, but we don't see those in the clinic. So, blue sky, how would you go about that kind of research, do you think, with the kind of infrastructure and opportunities we have at the university?
We've actually talked about this quite a bit at UC Merced about wouldn't it be great if we could enroll everyone, faculty, staff, students, as they move into the region and start on campus, and monitor them through questionnaires and to be able to find out when they get infected and look at that acute response. But we haven't been able to do that study.
So the people in Tucson actually looked at the students. Now they were symptomatic, so they looked at all the students who came to student health with symptoms of pneumonia, and they were interested in what percentage of those cases would be coccidiomycosis, and turned out to be about 25%, which is astoundingly high. So obviously in a highly endemic area, this wouldn't take very long to get a large number of people. Now, whether they'll be willing to let you poke them in all these different ways, that's another story.
I will say that the hospitals and clinicians in the region, including those in Kern County and then UCSF Fresno and Children's Hospital in Madera, have all been really open to providing samples and collaborating with us on these things. So we've been able to get samples through these endemic regions. So there's a lot of potential there, and I think the coccidioides field in general is very collaborative, and I think we have an opportunity to make a lot of headway.
Well, sounds promising. So thank you, Dr. Hoyer and Dr. Ferrer, for your participation here, and it was very informative, and we'll move on to the next co-presentation now.
Thank you.
Thank you.
So we want to now introduce Dr. Michael Peterson and Dr. George Thompson, who will do a co-presentation on the clinical aspects of valley fever. And just a reminder to everybody online that we will return to a moderated discussion later, so we can return to some of these questions later in the day as well. But now I want to introduce first Dr. Peterson, who is Chief of Medicine and Associate Dean at UC San Francisco Fresno. As a pulmonary and critical care physician, he is well versed in the challenges in diagnosing and treating valley fever in an endemic area. In 2009, Dr. Peterson developed the first multidisciplinary lung nodule clinic in the Central Valley, and he deals with the confounding diagnostic challenges presented by valley fever in this clinic on a weekly basis. He's going to be joined in this co-presentation by Dr. George Thompson, who is an associate professor in the departments of internal medicine in the Division of Infectious Diseases and the Department of Medical Microbiology and Immunology at UC Davis. Dr. Thompson is co-director of the UC Davis Center for Valley Fever, and his interests include not just coccidioidomycosis, but also aspergillosis and invasive candidiasis and other fungal infections. So thank you to Drs. Peterson and Thompson for your presentations.
Well, thank you very much. George and I will take on the task of outlining some of the many challenges that clinicians face around this disease. I think as John Taylor identified this, the unintended evolutionary host for this disease. So next slide, please. As several speakers have talked about, almost all cases of valley fever start as a primary lung infection through inhalation of the Arthrochonidia. And as others have mentioned, primary infection may be minimally symptomatic or may present as community-acquired pneumonia. Very difficult to differentiate from standard community-acquired pneumonia due to bacteria. I believe, as Dr. Ferrer mentioned, that in highly endemic areas, somewhere between a quarter and a third of patients presenting with community-acquired pneumonia may actually be due to cocci. And obviously, treating community-acquired pneumonia, which is due to cocci, with antibacterial antibiotics won't help the patient, and it really represents an example of inappropriate antibiotic exposure. It's also unclear whether early antifungal treatment for these patients would really alter the clinical course of their disease. The primary diagnostic tool we have available for differentiating the causes of community-acquired pneumonia is serology testing. And the problems with serology testing is that it takes time to get results back on that. It may take as long to get results back as the course of antibiotics that you put the patient on. and it may not be positive in early stages of the disease. Next slide. So some of the research needs that we see for valley fever that is presenting as community-acquired pneumonia is we really, to touch on some of the public health issues, need a better understanding of the epidemiology of community-acquired pneumonia, which is caused by valley fever in California. I think as Dr. Fearer mentioned, most of the epidemiologic data in this comes from Arizona, and the situation may or may not be similar in California. We clearly need better screening and early diagnostic tools for community-acquired pneumonia due to valley fever, and they need to be available to the clinician in a reasonable time frame. And these would be looking at, one of the things that we've been looking at is genetic markers, looking at PCR as a diagnostic tool in these patients, looking at more non-specific markers differentiating bacterial infections from non-bacterial infections, such as procalcitonin, potential studies for antigen detection, which have been done, but primarily with immunocompromised patient populations, and very little has been done in immunocompetent populations. Next slide, please. So the second aspect that I want to talk a little bit about is what happens after the primary pulmonary infection. So as several speakers have noted, most people will recover from this infection and have very few sequelae. However, one of the sequelae that can be completely asymptomatic is the development of lung nodules in these patients, and this may happen in as many as a third of those patients. They may also go on to develop chronic disease or dissemination, as others have spoken about. One of the challenges for lung specialists are the lung nodules. And what has happened over the last 5 years or so is the recognition that screening high-risk populations with CT scans for lung cancer is an effective way to reduce the mortality from lung cancer. However, unfortunately, a significant number of patients will have lung nodules found on lung cancer screening CT scans that are not due to lung cancer. And we anticipate that that number may be even higher in our region of the country. And as was noted earlier in the public health, there's increased incidence of valley fever, and that may also lead to more of these nodules in endemic areas. Next slide, please. So I wanted to demonstrate two representative CT scans that can show you the challenge that a pulmonary physician has viewing these patients. The chest CT scan on the left side is a lung nodule, which is a residual result of a primary infection with valley fever, while the lung nodule on the right is actually an early lung cancer. And as you can see, it would be incredibly difficult to look at those two CT scans and differentiate what it's due to, whether it's lung cancer or not. And without a history of coccidioidomycosis, so those patients that present with community-acquired pneumonia and are never diagnosed, or those patients that present with relatively asymptomatic disease, we don't really have a way to go back to see, well, are we developing nodules in an area where we had an acute infection, or is this something de novo? And that's where some of the difficulty arises. And this can really be difficult as a pulmonary physician then, and what we found is that we often have to go to invasive procedures or surgery to diagnose something that really at this point is a benign disease. So it's not just the morbidity and mortality related to the infection, but also the consequences of the infection and the cost and anxiety that that generates for the patient. Next slide, please. What we have found and others have reported is up to 30% of the incidental lung nodules that we are concerned enough about to send to biopsy in an endemic area is really due to valley fever. As I said, this leads to unnecessary expense, risk, and complications for the patients. The other side of the coin is waiting for longer diagnostic studies to come back to you means a delay in the possibility of diagnosing lung cancer. And we recognize that these delays, even in terms of weeks to months now, can actually negatively impact outcomes in the treatment for lung cancer. We have found in some of our research that serology is not sensitive sufficiently, and that PCR is also not sensitive for identifying those patients who have lung nodules due to valley fever. In preliminary research, we've also found that some of the decision support tools that have been developed internationally for helping the physician make decisions around which patients would require more invasive studies are highly inaccurate for nodules that are due to cocci, and does not really help us in terms of cutting down on those invasive studies. So one of the things that we need for this is both more accurate diagnostic tests as well as better decision support tools in our populations for patients from endemic areas. So at this point, I'd like to turn it over to George, who's going to talk a little more about some of the therapeutic implications.
Hi, good morning. I hope you guys can all hear me okay. So one of the real pressing questions we encounter on sort of the front lines of taking care of patients with this disease is we still don't really know what the best agent to treat them is. So traditionally we've used two different antifungals, fluconazole and itraconazole. Both have been around for quite some time, but the newer agents you can see on that second line, voriconazole, posaconazole, and isavuconazole, we really don't have a lot of data about how effective they are, if they're superior to other treatment options. The main limitation to using those agents is definitely price. These three newer agents are all more expensive. But we really do have the opportunity now with a number of new agents in development, some of which are now in phase 2 trials, to really determine what's the best agent for the treatment of this disease. There's been a lot of discussion about Nicomycin Z for a number of years. It's been around since the 70s and is still not available to our patients. But there's at least 3 new drugs that are all in phase 2 studies, one of which is Cidal. And what that means is it actually kills the fungus, even in the brain, in that animal study. So I think, and again, this is an orphan disease. They can do very small studies up to just 50 patients to get an indication to use these agents for the treatment of our patients with coccidioidomycosis. And I think positions us at the right time and place, maybe to even support the development of one of these agents through clinical trials, again, just in a very limited way with only up to 50 patients, and something that can easily be done in a relatively short amount of time to make these agents available for the treatment of our patients. So next slide, please. So one of the other things we really struggle with is how do we evaluate these patients that are going to go for a solid organ or bone marrow transplant. So the UC system has a huge number of patients that receive either a solid organ transplant, such as a kidney, a lung transplant, or liver, and then a very large bone marrow transplant population all throughout the UC system. The bone marrow transplant group at the UC system is already fairly collaborative and linked together, and I think we could partner with them easily to sort of develop a screening platform that's effective before they receive a bone marrow transplant. And the reason for that is sort of what was alluded to before. Once our patients are exposed to this fungus, they may deal with it well and sort of sequester it, but during periods of high immunosuppression, like when they get a bone marrow transplant or a solid organ, it can recur, it can come back. So how we identify those patients and adequately assess their risk, we're not very good at. We have sort of protocols we've developed. They've been studied in sort of at one center in Arizona, but I think validating those and sort of following those patients longitudinally is just really essential to make sure we actually know what we're doing rather than just think we know what we're doing. So I think that those consortiums can easily be leveraged for this. We also do need to do sort of a longitudinal study of patients that do have an infection. These are very immunosuppressed patients and have a high associated morbidity and mortality if they are infected with coccidioides. So sort of tweaking and establishing a better treatment paradigm is really essential for the care of those patients. So I think this, again, would have take a relatively short amount of time and have a big impact on our patients' care just over the next couple of years. Next slide, please. So the genetic risk factors, and this is something that's just been kind of near and dear to my heart. It's been a long-running question, of course, in the field, and Josh has done just some wonderful work on this over the years. And I totally agree with Katrina and him and John and Anita. This is really a fundamental question, sort of the host pathogen interaction. And so there's clear genetic risk factors for this disease. Men develop the disease more often than women. You could argue if that's just exposure or not. But the Filipino and African American patients are really just at a huge risk for disseminated infection. So that's complicated infection outside of the lungs. And so, some of the mouse models that Josh has worked on are listed here. And then some of the human patient mutations that Steve Holland at the NIH has found. And there's two concurrent efforts to look at this. One's an intramural extramural grant with the NIH that's looking at a bunch of these patients that we're sending to them. And another is sort of just patients that I've enrolled in this study by myself. So that's 500 patients we've acquired DNA from and sent off for whole exome sequencing. And so I bring that up not because I think that that's going to tell the answer, but no matter what the result is for that whole exome study, we're still going to need the studies that Josh and Katrina bring up. So if the answer to our whole genome analysis is we don't find anything, we're going to need a larger cohort. And if we find an answer that's postulated or likely to be implicated, we're still going to need to replicate that with the second cohort and prove it in an animal model. So I don't bring those up to say that we don't need the other work, but that we are definitely going to need other work regardless of these results. So next slide. So one other problem that's recently come up is really the susceptibility of the organism itself. So this had really never been looked at in a large-scale way. And what I mean by that is if you take coccidioides and grow it in a test tube or on a plate and then expose it to different antifungals, how susceptible is it? And you can see here we used over 400 different coccidioides isolates. And that kind of green graph on the left is amphotericin B, which is an intravenous-only medication we can't give for very long periods of time. But the next one over is fluconazole. And so you can see high numbers are bad on this. That means the higher the number, the more resistant it is in an in vitro setting. So you can see fluconazole really, even though it's the most prescribed antifungal, it has the highest MICs across the board compared to these other agents. So we may actually be using the wrong drug and the least effective drug. And I think that's also sort of anecdotally been proven when we know we need to use really high doses of fluconazole for efficacy in these patients. And we know from a single comparative study of fluconazole versus itraconazole was superior for those with bone disease, so those with real severe infection. So it's difficult to extrapolate something in vitro like this study really directly to the care of our patients, but it shows us two problems. Fluconazole is probably not the most effective, at least in vitro, and there's clearly resistance that does develop, and we've done quite a bit of work on that, and we think we know the mechanism, but we still really need to correlate that closely with patient outcomes. So that work is just about finished. But again, is going to need to be replicated in a big consensus way to really make broad generalizations for the treatment of our patients. So next slide. So that sort of summarizes the key questions that Michael and I thought would be addressable just over the next few years. There's obviously huge questions in the field that we really need the help of the whole community to answer. But just to sort of rehash this again, this problem with lung nodules, differentiating those from cancer is just a huge issue. The treatment, I still don't think we know exactly how to treat these patients with the best drug for the number of reasons we outlined. Transplant consortium would be very easy to do. And then the genetic predisposition, again, we're going to need to replicate that data no matter what we find from the other efforts that are ongoing. So thank you all for your time.
Thank you very much, Doctors Thompson and Peterson. That was very helpful presentation. We'll turn now to some questions.
Dr. Thompson, I just wanted to follow up on the last item you mentioned and how you need to really replicate in humans the idea of the pathogen susceptibility to the different drugs and understanding, if we're really using the right drug. Do you think that for that kind of question, one could leverage the electronic health records of the UC system to answer it?
Yeah, that's a good question. So there's two things I would bring up. So when we first just talked about this, I think a very big picture thing that really needs to be done is for all the UC system to kind of have these firewalls between systems, kind of at least have some holes in them for big research consortiums like this. I don't want to say bring the firewalls down because I think that's certainly dangerous. But this specific question I don't think is answerable because the vast majority of patients with coccidioides, their isolates aren't ever recovered. The diagnosis is made by a blood test or serologic method. And then even in the patients we do have an isolate recovered, they're very infrequently sent off for susceptibility testing. This is a really easy question to answer in a mouse. You know, the NIH has really supported drug development for a number of other fungal pathogens. And I know that they've really started and been very helpful with coccidioides support, but I think one of the NIH contracts to look at coccidioides is really key. And if they're just not able to do that, I think that's a really easy question for us to answer in just a year or two's time in a mouse model. We have all of these isolates in the lab and access to thousands of others as the UC system. So I think this is pretty easy to look at.
Thank you. So following up on that last point and some of the remarks that both you and Dr. Peterson made, I heard a lot of crossover comments regarding the intersections between clinical research and sort of host response mechanisms. And so you mentioned one, but what do you think are some of the other accomplishments that could be made, let's say, key research outcomes that could be successfully pursued in, say, a short-term 3-year period versus a longer-term research agenda of 10 years for both of you?
Well, let me touch on one of the things that Dr. Hoyer talked about, and that is the ability to obtain alveolar macrophages from patients with this disease. It's something that we can relatively easily do in pulmonary medicine to be able to provide her those kinds of cells to do some of the kind of research she's looking at, as well as cytokine profiles in those patients. So I think that whether we like it or not, we're a laboratory for this kind of research in that we have the patient population, and we'd be more than happy to work with investigators to provide them the clinical specimens that they need to ask the questions that they're looking at.
Dr. Thompson, do you have anything to add?
Yeah, I think if we could just identify a few of the genetic mutations that are really responsible for patients with very bad disease, that can very quickly sort of be leveraged into a screening test. And what I mean by that is if someone from Minnesota wanted to take a golf trip to Arizona, we could screen their genetics and see if they're at very high risk for dissemination and either put them on prophylaxis, antifungals while they're gone, or sort of tell them to take a vacation elsewhere. So the skin test is helpful to sort of look at that from a non-vacation standpoint. Our prisoners are at really high risk in the endemic region, and we do skin test them. The new skin test has some problems with it, but about 50% of the prisoners, regardless of the results of their skin test, they refuse to be allocated to a different prison system and insist on being stationed in one in the endemic region despite having a high-risk ethnicity. So, not all African Americans and not all Filipinos disseminate, just a significant number do. So if you could screen those patients genetically, that would also assist, reducing exposure to a big percent of those at risk as well. So again, and this is also really important, the work that Josh and Katrina bring up. That's the first question our patients ask us is, why did I get this? And we don't have an answer for that, despite this being a question, that was noticed in the 50s, I think, is when this sort of association with genetics was the ethnicity was first noticed. So that's a really key question that I think is going to take a great big group like this to answer.
Thank you. I want to get back to Dr. Peterson and your, really need for a fast diagnostic test, partly to start the right treatment, distinguishing between bacterial and fungal infections. And you mentioned, some of the things that could be done. What is your ideal diagnostic test look like? Like really, if you could do anything you wanted, what would you want the test to be like?
Well, so I think you're going to want a test that is fairly specific, easily run in a number of laboratories, doesn't have to go to a centralized or specialized lab, that you notice that some of the hot spots, if you will, for cocci are in parts of the state where there aren't large medical centers. So those patients are going to be presenting to their primary care physicians in that area, and if everything is going to require, sophisticated equipment and to be able to send to central labs, then you'll delay the ability to make the diagnosis in a timely fashion. So I think that those are the key elements about the patient population that's at greatest risk.
So we have a question here. We've heard a bit about the potential benefits of blockchain technology for both drug and vaccine development to manage the data across multiple institutions as research goes forward. And Dr. Thompson, you mentioned sort of the challenge of sort of being able to share data in electronic and protect privacy and also share data in our electronic medical records. So there is really here a question about how do we both share data in ways that can advance research, and also what are some of the challenges of that, so the benefits and challenges. I don't know if you have any thoughts about the possibility of incorporating blockchain technology or other thoughts about this question of data sharing across medical centers or research institutions.
Yeah, definitely. And I think that we don't need to reinvent the wheel, so to speak. So the UC system, so they sort of started with evaluation of deep venous thrombosis, or DVT. So that system is already built for the entire UC. And I think we really just need a green light from UCOP, to be totally honest, because I think pressure from sort of an administrative above would really help to sort of take the software that they develop for capture of DVTs and outcomes, which is much more nuanced than that, and we could extrapolate that to a lot of different areas. I mean, coccidioides is what we all care about on the phone today, but you know, the Cancer Consortium, the Leukemia Consortium, all of these groups would really benefit from that type of a system that's set up with just kind of a little bit of nudge and help from above, so to speak. So I think that would just really be useful to all of us. There's key questions in Cocci, but as somebody that sees patients with a lot of different fungal diseases, we need help in a lot of those different ways. Our leukemia doctors say the same thing. And you know, the UC system is bigger than Germany, so it's easy for us to make huge strides in a number of different diseases with just sort of a sharing ability to share data across the whole UC system with blockchain or something like that. So I'd be very excited about that, but.
Thank you. So maybe as the last, getting to the last question in this panel, I want to ask Dr. Peterson, you did mention the great challenges regarding lung nodules and you know, and the various issues that arise by having them. How do you actually manage in a day-to-day practice in an endemic area these lung nodules?
So we have a multidisciplinary team that sees these patients. We look at what the underlying risk factors are for this potentially being due to lung cancer. It's a joint decision-making process then about do we feel that the patients are at significant risk enough that we want to proceed to a biopsy of these lung nodules or not. That's the first step that's different than happens in the rest of the country, where if patients fall into a high enough category, you bypass the biopsy. But we found that if we do that, that we're taking a significant number of patients to surgery that have benign disease. So that's the first problem. The second problem is the use of metabolic scans like PET scanning, which again is utilized in most of the rest of the world to look at the risks that the lung nodules that we're looking at are due to lung cancer versus a more benign disease. And unfortunately, our experience in the data supports that Cocci nodules can have increased PET activity, so that doesn't work for us. The third thing is that there have been tools which have been developed where you basically plug in the characteristics of the patient and it assigns a risk factor to you. And again, we've looked at those in our patient population, and unfortunately in 85% of the patients where the nodules are due to Cocci, it still puts them into an intermediate to high risk category. So it becomes a lot of art of medicine to decide, do we feel that these patients really, because we're in an endemic area, that we can wait on or not. It makes it a very uncomfortable place as a clinician to be making those kinds of non-scientific decisions about a significant disease. And that's why we find that in a quarter to a third of our patients, we end up saying, let's just take them to biopsy because I don't want to take the risk, or the patient's uncomfortable with the risk of a watch and wait. So that's a lot of patients every year that we're sending to invasive procedures with cost and complications and so on, that it would be much better if we could recognize them and avoid that.
Thank you.
Yeah, that sounds like a very important subject to pursue. So I think I want to thank Dr. Peterson and Dr. Thompson for your presentations and contributions right now. And we'll see you and the other 4 UC presenters again in the final discussion. And so this does conclude now our research overview. Thank you again for all this, to all the 6 speakers. Prior to the final moderated discussion, in this next part of the webinar, we will now consider how UC and our researchers can have the greatest potential impact on valley fever for research. And so we will next hear from important contributors and collaborators, potential collaborators on the state and federal level about the work that they are doing to advance progress on this disease. I'd like again to let all the webinar, I'd like to let all the webinar attendees know that during the next 3 presentations, we will not have a Q&A period after each speaker, but we will pick up questions in our final moderated discussion, which will include these speakers as well. So please continue to submit questions during this portion of the webinar, and we can get back to those questions during the final discussion. So I'd now like to introduce Braden Murphy. Mr. Murphy is Legislative Assistant to Congressman Kevin McCarthy, the U.S. Representative for California's 23rd Congressional District that includes the Southern Joaquin Valley and Bakersfield, a region particularly impacted by valley fever, as we've heard. Mr. Murphy will tell us about the federal forward legislation that is sponsored by Congressman McCarthy.
Excellent, excellent. Hello, everyone. Thanks for having me. As indicated, I'm Braden Murphy. I handle the healthcare portfolio for Congressman Kevin McCarthy, whose district encompasses the southern part of the Central Valley, largely Kern County, which, as you know, routinely reports the highest number of cases in the state. I believe in 2017, it was 37% of all cases statewide. So more or less the next 5 minutes, I'm just going to give you a very brief federal overview, history, and update on what we're doing and what he's done since he's made this one of his top priorities since coming to Congress in 2007. So I'm going to go ahead and back up 5 years. Essentially in 2013, Congressman McCarthy organized and put on the Bakersfield Valley Fever Symposium, where he invited the NIH Director Collins and then CDC Director Frieden to listen to patients, discuss research, and really truly shine a light on valley fever. It's the first of its kind, and it really moved the ball forward for the disease. And resulting from that symposium was the NIH-sponsored randomized control trial to test early treatment of fluconazole on valley fever. Unfortunately, there's been some challenges with enrolling patients, and earlier this year, patient enrollment's been put on hold while they redesigned the trial. But I think once the trial's redesigned and approved and patient enrollment is ultimately resumed, I think it's going to be critical to spread awareness within the public and in the broader medical community to spread word of this trial that's going on to help maximize the efficacy. Moving forward, this year in July, the Congressman introduced the Bipartisan Forward Act, which stands for Finding Orphan Disease Remedies with Antifungal Research and Development. And this was truly the product of months of calls with mycologists, clinicians, patients. It was a very collaborative process, and it's 28 pages. I won't bore you with the details, but I'm going to highlight some of the provisions here that I think are most pertinent for this webinar. One of the provisions, it creates a federal advisory committee, a working group consisting of representatives from the FDA, from the CDC, from the NIH, then also mycologists, researchers, clinicians, patients, and they're tasked with essentially meeting annually discussing the state of play in terms of research, where there are any gaps, and publishing a report to kind of create a comprehensive approach to where we can improve and address this issue. One of the main provisions I think that's most pertinent to this webinar is the endemic fungal disease state match provision, which essentially, as has been highlighted in this webinar, the state recently came forward with $3 million to the UCs, $3 million to Kern Medical's Valley Fever Institute. And essentially what this provision would do here is allow those entities, nonprofit, university, hospital that had received state funding for endemic fungal disease research to essentially be able to apply competitively for a federal match to that state funding. And the rationale here was, endemic diseases largely don't receive as much resources or attention as those that affect folks from all different corners of the country. And so this, this provision aims to more or less encourage states to continue to pony up more or less and dedicate resources to diseases that largely impact their citizens. So I think that's something that'll be very, very important moving forward. I guess the other provisions of the bill, and I might get into too much detail here, but essentially they aim to incentivize industry and antifungal development. And I'm not a mycologist, I'm not a scientist, I'm barely a paper pusher here in DC. But the moral of the story from what I've heard after all the discussions with mycologists and researchers is that developing antifungals is inherently complex. And so some of what these provisions do, one of them would direct the FDA to issue guidance for industry after considering stakeholder input to essentially convey FDA's thinking on how and what the best way to get antifungal drugs, diagnostics, treatments approved. There are other market incentives, largely the extending GAIN Act, generating antibiotic incentives now, was passed in 2012, I believe. This would extend essentially it's market exclusivity, priority review, and eligible for fast track review at the FDA. And this bill would extend to antifungal biologics. So for a vaccine, potentially a Valley fever vaccine, they would have additional market exclusivity, priority review, so the FDA responds to a new drug application within 6 months, and fast track, basically enhanced coordination and collaboration with the FDA early on. So that was more or less aimed at trying to incentivize Valley fever vaccine development. There's also the priority review voucher program, which would essentially, the priority review voucher program exists for rare pediatric, tropical diseases, and more recently medical countermeasures. And essentially what it does is if you get a new drug approved for, say, a rare pediatric disease, you get a priority review voucher, which you can either use for yourself, use within another drug application within the company, or you can sell to another company. And this would essentially extend that existing program to endemic fungal diseases. There's many other provisions in that bill. It's 28 pages. I encourage you to take a look at it if you have time. It's referred to the House Energy and Commerce Committee, where we're working with committee staff to move it forward. No pun intended on the name there. But I also wanted to highlight some other things that are going on the federal level. There's the Congressional Valley Fever Task Force, which is comprised of lawmakers who represent endemic regions, so California, Arizona, Utah. And they work together to advance Valley fever priorities. So earlier this year, Congressman McCarthy led a letter with 17 of his colleagues in the House in support of an NIH SBIR grant to bring quicker Valley fever diagnostics to primary care settings. That's still being reviewed by NIH, but it's a great working group that is specifically focused on Valley fever on the federal level. That's more or less it. Again, I think I'm running out of time, but thank you for having me again today. If you have any questions, if you'd like to voice your support for this legislation, or if you have thoughts on what we can do further on the federal level, please give me a call or give the office a call, I guess, and I'm always happy to chat. So, thank you.
Thank you so much, Braden, for your remarks. And I think, I think I speak for UC and all Californians really that are concerned and affected by valley fever, about how grateful we are for these efforts on behalf of Congressman McCarthy and his some of his colleagues at the federal level to address this important public health issue, and to think about the role of research and the role that the University of California also can play in making a difference in this area. So thank you for your time joining us today. And we now want to introduce our next speaker, who is Dr. Royce Johnson. Dr. Johnson is the medical director of the Valley Fever Institute at Kern Medical, and he is also an adjunct faculty member at UCLA. Kern Medical is a recipient of $3 million in state funding that Assemblyman Salas mentioned earlier today, so that Kern Medical can also address valley fever. And Dr. Johnson will tell us a little bit more about how the Valley Fever Institute at Kern Medical will be using these funds and working on the state initiative. Thank you.
Thank you very much for having me today. It's a pleasure to participate. I've certainly been impressed with both the people that I've known well for years and the other people that I've not known that have presented this morning. I will also want to take a quick moment to thank Senator Fuller, Assemblyman Fong, Assemblyman Salas, who of course presented, as well as Kevin McCarthy and his office and staff for their past and future contributions to trying to solve this disease, especially since I live in perhaps the most impacted area with valley fever, other than possibly Tucson, Arizona. So the Valley Fever Institute wouldn't really have a mission if we didn't have the money. And for that, I am really grateful. I'll briefly outline what I think our mission is. I want to partner with public health, both state and local, perhaps national, and the Valley Fever Foundation of the Americas for public education. Knowledge is power. We're also committed to developing and doing professional education, epidemiology, diagnosis, treatment, and follow-up, which people often don't talk about, but appears to be a major problem. We want to develop a coccidioidomycosis patient-centered home for improved care of our patients and a model for others. So we want to have quality care, psychosocial support, clinical pharmacy, patient navigators, infusion center, a center for intrathecal amphotericin for meningitis patients that fail azoles, which continues to be a problem. We want to be involved with clinical studies, perhaps phase 2 with nicomycin Z. We're involved with the NIH Duke effort on fluconazole, which is having a new iteration. We want to develop a database. We already have actually a database of patients with almost 2,000 patients in it, which can be mined for clinical research. We want to develop, since we have more cocci patients than all of the rest of you that have spoken combined, a repository of serum and cells that could be studied in other people's laboratories, as well as with my colleagues at UCLA, Paul Krogstad, Manish Gupta, and Wayne Grody, period. I think the best use of this money is in the strength of UC, which is the ability to do high-level immunogenetics and genetics in terms of the $3 million that go to UC. Thank you very much for allowing this time.
Thank you, Dr. Johnson, for sharing your vision, really, for how to advance care and care for patients and combat of valley fever in your highly affected area. Our third speaker now in this segment of the webinar is Dr. Duke Vuja. He's the chief of the infectious disease branch at the California Department of Public Health. CDPH received $2 million in the state budget this year for valley fever education and outreach. So Dr. Vuja will tell us about their goals and activities for this funding.
Thank you.
I don't see my, can you hear me? Yes. Okay. I don't see my, okay. I'll go ahead and speak since you can hear me. Thank you for having me and good morning. You've heard from Assemblyman Rudy Salas earlier, and he and his colleagues had passed 3 assembly bills this year on valley fever. One of which is, I was just mentioned briefly, is Assembly Bill 1790 that requires, it's basically called the Valley Fever Education, Early Diagnosis and Treatment Act, to raise awareness of the symptoms, tests, and treatments for valley fever among the general public, primary healthcare providers, and providers who care for persons at higher risk for valley fever. The bill requires us in the California Department of Public Health to conduct a valley fever awareness campaign to communicate with local health jurisdictions, providers, and the public about valley fever, to include information about valley fever symptoms, diagnosis, and treatments, and to provide outreach to the general public, endemic regions, and at-risk populations, healthcare professional associations or societies, healthcare employers, and local public health officers and health departments. So we were happy to know that in the Budget Act of this year, that the California Department of Public Health also received $2 million to basically implement this awareness campaign on valley fever. The funding is available now through June 2020. Now, up until now, we in the department has been conducting education and raising awareness on valley fever in our limited capacity, but we have provided, we have set up a website and for many years have provided posters, brochures, and fact sheets on valley fever, both for the general public as well as for high-risk groups in English, in Spanish, and Tagalog. We had also put on the website information for healthcare professionals. We have provided brief updates on valley fever to all licensed California providers in the California Medical Board newsletter periodically. And this recent month in August, the Valley Fever Awareness Month, we have worked to put out social media messaging. And our occupational health branch has also conducted a webinar on work-related valley fever. So we have been doing what we could with our capacity, but now with the additional funding in the budget, we will be planning to use this funding to plan for a broader, larger campaign of valley fever awareness. We're going to be hiring an a valley fever awareness campaign coordinator. We'll be looking to contract with a media marketing firm to plan and implement all of this with us. And basically it's going to be a statewide campaign for the general public, for those people living in areas with increased incidence of valley fever, targeting also populations at risk for severe disease, including older adults, pregnant women, African American, Filipinos, and persons who have who are immune compromised. We'll also be targeting healthcare providers, hopefully providing not only health educational materials, but also continuing medical education. And finally, we also will be working with employers of workers who may be at risk for valley fever. So currently, we have started this process and are working with our local partners, local public health and other partners. And then as a last pitch, we do have some epidemiology resources in my branch that we're more than happy to leverage and work with any of you who would like to collaborate in our capacity. Thank you.
Thank you, Dr. Buja. That was very helpful, and also Dr. Johnson and Mr. Murphy for all of your comments. We are grateful for your comments, Dr. Buja, on that opportunity to collaborate and leverage, because I do think that that is one of the strengths of the University of California, is not only the ability to work across the multiple campuses and medical centers here at UC, but also to reach across and work with all of our state partners. So we're going to shift now to a moderated discussion, and we wanted to, at this time, invite the presenters that just spoke, the 3 presenters that we just heard from, together with the 6 UC presenters, to turn their cameras back on. For those of you watching, it's going to look something like what we're calling Hollywood Squares, if you're old enough to remember that. And while their cameras are coming back on, we will also remind attendees to please go ahead and look at the poll questions that we are now going to project on the screen. This is an opportunity for us to collect some just high-level responses from our attendees today. And if you'll just take a moment to read those 2 questions that we're posing to you, and then we're going to come back to those results after we've talked a little bit more to our presenters. So with the help of our folks here, Uta and I are going to continue to pose some questions to our speakers for most of the remainder of the webinar today. I wanted to just take a moment to see if there are any particular comments that Dr. Johnson and Dr. Buja both mentioned, sort of opportunities to work together with the University of California, and if there are particular opportunities that you see beyond those you've already remarked on that we could really target in this this first initial period. Dr. Johnson, your audio is off.
Is it on now? Can you hear me now?
Yes.
Yes.
We really are at Kern Medical, the University of California, and the Department of Medicine, and we do collaborate with our colleagues at UCLA, and I've also collaborated significantly with George Thompson on various projects. So there is a nascent ability to collaborate, and I think George very nicely pointed out that this could be expanded in a substantial way.
Thank you. And Dr. Buja, you mentioned epidemiology research. Is there any other areas or opportunities you think are particularly stand out?
Well, our strength is epidemiology as we conduct surveillance for cocci throughout the state. And we have actually collaborated with various groups and people before. For example, Royce, we've worked with Dr. Johnson in the past on looking at specific groups of patients in Kern. With the CDC. We also work closely with CDC in terms of the broader picture. Our strength in using the epi data that we collect on patient is basically to look at risk groups in general, areas where there's moderate to high rates of disease. And if that contribute or facilitate any of the studies that any UC researchers are doing, then we'll be happy to provide information or work with them on it.
Thank you.
So I'd like to continue thinking about what it will take to really move this field forward rapidly. And we've heard in prior discussions from some faculty researchers that one of the key needs for valley fever research is training a new generation of scholars who have the tools to pursue new breakthroughs. And so the question that I'm going to pose, and it probably might be very different answers for the different stages of the pipeline of basic science to clinical work, but the question I'd like to pose is, to what extent do you see the career pipeline as an important challenge in your area? Maybe we can start with Dr. Taylor and Dr. Sill to reflect on the needs for filling that career pipeline.
John, do you want to start, since you've been, I think, instrumental in recruiting people to the basic science of coccidioides?
Well, I think that it is very important to have a pipeline of young researchers who are going to become interested in researching cocci and the disease. And if you look in Arizona and California, there's an older generation of researchers that look like me, and then a younger generation that look like Anita. And this is throughout California. Certainly UC Merced, UC Riverside, Northern Arizona, And I even put George Thompson in the young group at UC Davis, that there are young people coming up. But to get young people interested in a field, there have to be resources. And so the work that Assembly Person Salas and Representative McCarthy are doing to bring resources to the field of the study of cocci are essential. Without those, there will be no new generation.
And I would just add, you know, one of the limitations in a basic science laboratory for pushing projects forward is essentially finding the funding for students and postdoctoral fellows. And that is that would be enormous in terms of our very effective, but, you know, a little bit, you know, but visionary types of approaches.
Yeah, maybe I'd like to now, you know, sort of go into the next step of, I saw, I think Dr. Johnson, I might want to, actually Dr. Johnson, if you wanted to comment.
Yeah, I certainly agree with what John and Anita said, but I also have concerns about the fact that there are probably less clinicians interested in this disease now than when I entered the field, I am embarrassed to say, 40 years ago. So we're not raising up a younger generation of clinicians in various and sundry disciplines that are interested in the care and evaluation of patients with cocci and doing clinical research on them. I actually see that basic science has moved ahead of where it was a long time ago, but that clinical care and science has lagged.
Oh, very, very interesting. We'll get back to immunogenetics, but maybe to keep this thread going, Dr. Peterson, did you want to comment on that? And Dr. Thompson?
Well, I would agree that within sort of two areas. One is that all of the infectious disease physicians on the panel today recognize the challenges in bringing physicians into the field of infectious diseases. It's been a real challenge over the last couple of years to be able to do that just in terms of numbers. And then I think it's the ability that, just as with the basic science, to be able to support people to develop the skills as a translational or clinical scientist to be able to do the work in an environment where more and more of the demands are there to do clinical work, not do investigative work.
Mm-hmm. Seems like Dr. Thompson is agreeing. So I'd like now to give Dr. Hoyer and Dr. Fearer an opportunity to also comment on the career pipeline.
So I would say with the career pipeline, I've really seen at UC Merced a great excitement by our students to be involved in outreach and research in Valley fever. Many of them grew up in the endemic region. And so we've had students kind of come out of the woodwork who are interested in participating. So, and I think a lot of our students are pre-med, and so there might be an opportunity for continuing to engage them in the clinical pipeline, as well as the basic science side.
So one of the limiting things, because this is a BSL-3 organism, is the availability of BSL-3 laboratories. And people have said that there are a lot of them. In fact, there are just a handful. The second is it's really a biohazardous organism. That's why it's a BSL-3. And frankly, a lot of people in the highest risk groups don't want to go into this research.
Okay, thank you. Yeah, I think that was very informative for us to think through, what are important needs in the field. Thank you.
And what about, now that we have all of you on the screen together, we've heard you talk about the various stages, and actually I thought it was very helpful the way that you referred back and forth to each other, all of you really, in your presentations, thinking again about collaborative research or interdisciplinary or even translational research. What do you think might be to what extent do you think that kind of cross collaboration at sort of at this point could be effective? If there is a gap on the clinical side, is that something that should have a higher priority, or is it really the linkage across the span that could be the most effective? Dr. Thompson.
Yeah, I think that those issues were addressed by colleagues, but so 30% of all infectious disease doctors are predicted to retire over the next 5 years, which is really going to be an even larger problem going forward. I mean, infectious diseases by itself has a lot of financial barriers for recruitment, but that's been very important to us at Davis, and I know a number of the other centers really have recruited younger faculty, trying to get them interested in coccidioides. That was a big focus at sort of the Cocci study group 2 years ago, is who are you mentoring? And I think that was recognized as a big problem in the field. And again, there's lots of issues for recruitment for that. I don't know we're going to solve all those today, but I think that in the spirit of this grant, really an emphasis on trainees in some fashion is going to be essential.
And could I comment?
Yes,
on sort of the opportunity here to link fields? I think it's immense. I mean, we are so lucky in the UC system to have the clinical strengths, the strengths in immunology, and the strengths in mycology that we have. And Josh is right that we're limited in terms of BSL-3 facilities, but there are people who run them who are interested in studying this organism. So it seems like there's tremendous potential for having a very, the organism itself is linking things for us, from the environmental, ecology to infection, the fungus itself and the host response. And in this day and age of collaboration and bringing, new technologies to study medical and scientific problems, it seems to me that there are obvious, collaborations where strengths in things like immunoprofiling and, modern ways to look at, to use CyTOF mass spec approaches to look at immune cells and the tissues that folks that interact with patients have, and the fact that we could potentially culture multiple clinical strains of the organism and profile them in a molecular manner. There's tremendous potential here for collaboration.
Thank you.
So thank you. I did want to encourage all the attendees to please respond to the poll. While we're still, going to continue the discussion, but later in this discussion, we'll reveal the poll results and comment on them. And so if you please could all answer the poll, that would be fabulous. We do have a specific question that Dr. Wuser can probably answer, and that regards whether the military has done any additional studies of cocci incidence in California.
I know that they've done, and there's been quite a few studies that were published in the 40s and the 50s, but I'm not aware of any at the moment. I know that there's some military epidemiologists who are interested in cocci, and we occasionally have crossed paths, but I'm not aware of any current study by the military.
There are a lot of clinical studies that are doing, especially at the Naval Hospital in San Diego.
So throughout the day today, we've also heard some comments and remarks on a couple of the presentations about differently affected groups and vulnerable populations. Some of them related to some of the genetic factors, and some of them from the earlier presentation also related to occupational exposures, prison exposure, and things like that. So I just had a general question about your thoughts on, given the difference between exposure and illness, and then the disseminated illness, what do you think are sort of, in terms of making a practical impact, are the relative benefits of looking sort of broadly at some of these genetic and immunological responses and clinical presentations as compared to research that might focus on particular groups at higher risk?
Well,
Dr. Johnson.
So this point about impacted groups caught my attention many, many years ago. And I was interested in the idea of immunogenetics and mechanism of disease. Because of seeing that, it was clear to me that there had to be a genetic underpinning to this. And we in fact did some studies on differences in MHC in people that I published some long years ago. But the genetic tools and the immunologic tools didn't exist to really study this in any organized fashion until relatively recently. But clearly, I think that studying African Americans is likely to be highly productive in terms of understanding immunogenetics. Clearly, not all African Americans are at high risk, so we use race as a surrogate for what is clearly, in my mind, an immunogenetic problem. In fact, my hypothesis about all of COXI, with all due respect to John Taylor and Anita, is that it is a genetic disease with fungal implications.
And Dr. Taylor, I saw that you were going to respond, and now it's a good segue.
Well, I think that there are two routes to dealing with disease that the NIH has, and one is treatment and the other is prevention. And so to me, missing from the list of ways to go forward is the prevention angle. And you know, the old saw that an ounce of prevention is worth a pound of cure is true. If you can persuade people or inform people so that they take precautions when they're in an area of high risk, or if they have a genotype that puts them at high risk, you would avoid a lot of expense. And with the present drugs, in most cases, a lifelong dependency on a drug that has side effects to control the disease. And so what we need then are better ways of assessing the environment and also better ways of protecting. And George Thompson raised a very interesting one, and that would be the prophylactic use of antifungal drugs. I don't believe there's been any test on that. And there are, I think George has told me 80% of the firefighters in California are prisoners. And they don't do the high-tech firefighting with hoses, they do the hard firefighting with shovels. And they then, they're disturbing the soil in areas where it hasn't been disturbed before. They're very high risk. That might be a group of people who would benefit from having one of the azole drugs or a different drug in their system during that exposure, or have some kind of mechanical protection to keep them from breathing the spore. So I agree with what everybody said about basic research, about the understanding of the human genome and susceptibility. And I don't dispute Royce's idea that human genetics are really important. However, I will note that if you get an overwhelming inoculum, even the person with the best genome will get COXI, has been demonstrated in lab accidents. And it's probably demonstrated when you put your shovel into the carcass of a dead mammal when you're out fighting a fire.
Thank you. Dr. Thompson, did you want to add anything to that?
No, I totally agree. There's some groups that we can't just say avoid the disease, and the prisoners that fight fires are certainly one of those populations. We heard about the solar farm workers. Those are groups that are at very high risk for very long periods of time. And wearing protective gear is just not an option for some of those patients because they're just in the environment for too long. They can't wear an N95 mask on 100-degree days for 12 hours in a row. So I think that it would be really nice to do a very small prophylaxis study really with our partners in public health in that particular population. I think that's a group, again, at really high risk. We know they're in the right area to get COXI. They can't protect themselves. So trying to help them in some of this smaller scale fashion is very easy to do. It's pretty low hanging fruit with the assistance of our public health colleagues.
Dr. Vuja.
Yes, an important tool in prevention, of course, is a vaccine. I was hoping to hear some comment or interest in research in further the vaccine development projects that were actually partially funded in the past and had start and stop kind of process. So I'm not sure whether, of course, it's a long-term commitment as well as in terms of both funding and resources and personnel. But I was wondering whether there's any interest in restarting any look or research, at least to start into the vaccine for valley fever.
Dr. Thompson?
Yeah, there's two groups, of course, that have vaccines in various stages of development. One's the University of Arizona group, and the other in Texas. The Texas group just published an article, I think, last week in Infection and Immunity, looking at their vaccine, which really has three components in an animal model. And then the U of A Arizona folks are starting their vaccine studies in dogs. So they've made some headway, but I think there's still a number of years to go.
There are some major practical problems with developing a vaccine for cocci. It's hard to bring vaccines to market. There's liability associated with that, but mostly the issue of who's going to pay for it. And frankly, there aren't enough people at risk for this to ever be a profitable thing. So it would have to be underwritten by some government entity.
Well, I just want to say, this is Duke again, and that's why I was hoping, I'm going to read the forward legislation that's being discussed at the federal level, because it does, I agree, it needs a federal commitment and support for this to happen. But I do believe that the populations that may be affected out there is large enough. My God, the entire southwestern United States is at risk as far as I'm concerned. And anyone who's going to be, snowbirding from other parts of the country to the southwestern United States to either retire or to spend the winter is going to be at risk. So I think there is a large enough population that could benefit from the vaccine. But you're right, we need federal commitment as well as long-term commitment to see this to fruition.
Dr. Johnson?
As the only one here that participated in Cocci Vaccine 1, which was done from 1979 to 1986, we ended up with a whole spheroidal vaccine with 9 in the placebo group and 9 in the vaccine group, and that's with a total population of 5,000 that were enrolled. About half of those were my enrollees. It took an enormous amount of effort. And the biggest problem with getting a cocci vaccine group or study, in my mind, is not actually even developing the vaccine, but doing a phase 3 study with a disease that has an incidence between, at least in average years, might be higher this year, good year for a vaccine study, is somewhere around 1 to 3% of the population become infected, and most of those aren't even symptomatic. So to prove that a vaccine works takes an enormous N to enroll, to study it. So it's going to be an extraordinarily expensive product to bring forward. On the other hand, the amount of money we spend caring for people with this disease is much more enormous. So I think it would be cost effective to do this enormous project. But again, the only person that could fund it would be the federal government. And the $3 million that we are talking about today would be a mere drop in the bucket.
Thank you. So one question I wanted to, we didn't spend as much time on today, is really about something you just mentioned, Dr. Johnson, which is that the disease outbreak related to valley fever is uneven year to year, and there does seem to be perhaps some environmental or climate effects, as well as perhaps the life cycle of the fungus itself. And I'm wondering if some of our speakers might want to talk a little bit about this question of the environment and climate change or warming and drying climate issues that are affecting California and the Southwest and other affected regions, and how you see that kind of research that kind of climate research intersecting potentially with valley fever.
Sure.
Does anybody want to?
Let me take the first stab. The first thing here is that this is a wonderful opportunity for the University of California to partner with the California State Department of Health and the Centers for Disease Control in Atlanta, because both groups have epidemiologists and they're very interested in modeling and understanding how all sorts of environmental parameters can affect the incidence of coccidioidomycosis in a given year. So prediction could be important for allocation of public resources for treatment that's going to follow these changes. And also these, the university and the Department of Health and the CDC could certainly partner together in looking at specific environmental parameters to help you understand which areas would be the most dangerous areas in which to disturb previously undisturbed soil. And climate change, as it gets warmer and drier in California, coccidioidomycosis is going to move north. No way around it, because the small mammals are going to move north.
I mean, do you actually see an opportunity to model this in a way that, you know, a public health strategy could be developed, understanding where and when physicians and healthcare providers need to be alerted, you know, to these changes?
Yeah, absolutely. And you know, models, I mean, one of the great things about models is they tell you what data you don't have and that you need. And so models, you know, you build a model and then you realize there's a gap in our knowledge here. And so then you need to add more data. And those could be, for example, the incidence of people with genotypes that make them susceptible to cocci, the number of people whose immune systems make them susceptible to cocci, and also all the environmental parameters that encourage the presence of Coccidioides species in the environment. And those could be used both to plan on treatment, but also to plan on the sort of prevention that you need in various areas.
Okay, thank you.
Dr. Hoyer, did you want to add something? No.
To go back to leveraging potentially, you know, the resources that are being provided to UC through the state funding at UC and others through the state funding, looking across UC, but also the state and other regional partners, including Arizona and Mexico, for instance. Do you see any sort of unique, important opportunities for collaborations there? That, you know, that could be, you know, really pursued and supported, or, you know, really perhaps by the funders, if we knew about opportunities to see.
Let me say one little thing. So the University of California Office of the President is funding some coccidioides research right now. In fact, Anita at UCSF, the group at Merced, some researchers at Riverside, Josh at San Diego are involved in a small project. And we have reached out to researchers in Arizona and are working with them on the project. And there's no reason that we couldn't reach out. In fact, researchers in Arizona have reached out to researchers in Mexico, both in Baja California and in other parts of Mexico. So having a regional look at it is an excellent idea. And researchers in Mexico have done some things that we have not thought of. So it's definitely good to get as many minds as possible into the operation.
Thank you. Yeah, yes, our office is supporting that project. So we're glad that it's been a help in addressing this issue, and also perhaps positioning some of our researchers to work together collaboratively in a multi-campus and interdisciplinary way. I wanted to return to a question.
So I think that there are two potential occupational groups that would clearly be interested in this, and these are the occupations that are at highest risk. So that is the prison workers and the agribusiness. So in terms of looking for partners that would potentially be interested in putting additional resources into that, those would seem like two appropriate places to look.
Thank you. I wanted to return to one of the questions we had submitted a little bit earlier, and I believe it goes back to either Dr. Sill or Dr. Hoyer's slides, but maybe both of you are, of course, welcome, or others to respond. Thinking about the CRISPR, you mentioned, there was mention of the CRISPR technology and other new generation gene technology, gene knockout technologies. Can you describe a little bit more about what's already been done with these technologies in relationship to COXI?
Yes, so the standard, what's been done in the field in terms of gene disruption in coccidioides is using agrobacterium-mediated transformation to generate mutants. In terms of CRISPR, which I mentioned in my slides as being sort of the newest, one of the newest tools for genome editing. So there are two labs that I know of that are trying to make CRISPR technology work in coccidioides, and that would be Aaron Hernday at UC Merced and then my lab at UCSF. And we have, so Bruce Klein's lab at University of Wisconsin, Madison, so there's a group of fungi that are closely related. Coccidioides, I'm just going to talk about three: coccidioides, histoplasma, blastomyces. Bruce Klein's lab has made CRISPR technology work in blastomyces. My lab has adapted that for histoplasma, and it's working extremely efficiently. And so we are trying to transfer those technologies to coccidioides now. And that would enable us, hopefully, to make mutations in fungal strains with greater efficacy.
Great.
I want to share a comment from our attendees. It goes back to the previous question around broader research collaborations and issues related to that, and that is a major public health issue. Apparently, that serologic diagnostics are not available in Mexico at this time. So I don't know if anyone could comment on that, but I think it was just a comment to keep in mind. Dr. Johnson?
I don't believe that certainly was true. We've had collaboration, a lot of it because of Tony Catanzaro from UCSD with Mexico for some time in the COXI study group. And I was more or less horrified in the early days of that participation that almost all the studies that were presented from Mexico were in fact autopsy series. But they've come a long way. They have a skin test antigen, and they do have serology in Mexico. That's a fairly recent innovation. I can't tell you the years. Maybe Josh or George or somebody knows, but they do. So they've come a long way, and they do have decent clinical research and some molecular research. And I think that this is a fruitful area for if we wanted to enlarge beyond UC or beyond California and Arizona. Collaborating with our Mexican colleagues would potentially be useful, probably logistically more problematic.
So I don't know what the current situation is, but a few years ago, my contacts in Tijuana told me that they had to send their serologies to Mexico City and wait for the answers to come back. So serologic tests were available, but not easily. By now, that may have changed.
We do, the University of California does have a partnership with a number of Mexican research institutions, and there may be some things that we can look into going forward for collaboration. I know the president and our office, Vice President Ellis, have been very interested in that kind of regional research partnership as well as with some of our southwestern states, Arizona and others. So I think there's some things there that we can certainly look at in the future. I had one other question, I think, before we move on, for Dr. Hoyer. I know that when we spoke previously to today, and I'll ask you to recall that conversation, you mentioned really a broad range of research that's going on at Merced that spans not just the sort of basic science research, but also in the Health Sciences Institute that also includes some community research and other kinds of research. And I wondered if you just wanted to take a moment to share some of the other multidisciplinary kinds of research that you're aware of there.
Mm-hmm. So there, like I said, there are 3 of us that are doing basic research with a UCLP grant with several other UCs that's currently led by Anita Sill. But we also have several researchers in engineering who are interested in doing some detection work and measurements, both in the environment and in clinical samples. We have several researchers who are they're reaching out with rural farm workers and local physicians to do outreach and kind of communication of this disease. We also have several undergrad groups that are doing outreach into various patient groups and different ethnic groups within the region to communicate the disease. So yeah, we have kind of all levels. We have a couple of our Spanish undergrad classes that are doing outreach into their communities. We have, just kind of all over the place. Lots of different researchers on campus. As I said, we have many of our students are from the region. They grew up with this disease. Their parents have had this disease. And so they're hyper aware of the research and the impact on the region. So they've all been very excited to participate. And that excitement has also spread to those of us that are new to the region and have started to move into this area. On top of that, as I said, this field has been particularly welcoming as a scientific community to new researchers and helping us learn the field and become active participants in the research that's happening.
Thank you. And I did. It does. Thank you. And I brought it up partly because I know today we did not have an opportunity to invite all of our UC experts to present during this webinar, but we are aware that there are a lot of individual researchers and research teams in many disciplines that are working on valley fever. And we know some folks are listening in today, and we wanted to make sure that we used our time here to focus on a few areas, but we know that there are many other areas and many research topics that we didn't have time for today. With that in mind, we wanted to show the results of the poll that we took earlier at the beginning of this moderated segment, so that people can see our presenters as well as us here can see how people voted on the 2 questions that we asked. So having heard all of the discussions today, what are the 3 research areas most likely to produce significant discovery to address valley fever in a 2 to 3 year period? And we got a lot of enthusiasm, the highest results in clinical diagnostic tools, but also climate change and environmental effects and host immunogenetics were the top 3 that came through. So that's very helpful, but for folks that can see the results, you can also see there's a wide range of interest across all of the topics that we polled. And then given UC's current infrastructure and expertise, as we were able to communicate it today, and of course, as I said, that we weren't able to represent everybody's research, but given what we were able to share today and what people are aware of, how might a $3 million investment have the biggest impact? And there was a lot of interest in seeding individual research projects across a range of topics. That was the 39%, so a lot of folks want to do that. And then some sort of equivalent levels of interest in collaboration and student and postdoctoral fellowships and research infrastructure. So we just wanted to take a moment to ask our panelists, especially those of you at UC, but others as well, if you had any comments or thoughts about these results. Can you also see them on your screens, the poll results? Yes. Okay. So given the some of the responses we had to training graduate students, but also great level of interest in seeding individual projects. Do you have any thoughts or comments you want to share with us on how we might think about best providing funding?
Well, I can comment on the student fellowships. I mean, student funding is always a really fantastic thing and is helpful, but without the research money to do the research, I think it really needs to be in combination, because you have to have the money to be able to ask the scientific questions in order to be able to then fund a graduate student to do that work.
So would you think that sort of a multi-year faculty grants that provide sufficient funding to bring on and train and engage the graduate students, is that what you're thinking?
I think if you're going to fund the research side, the students, I think that's what you have to do, is it needs to be in combination.
I've got a comment about it. I think that you do want to train graduate students. You also want to train postdocs. My experience is that if you train a postdoc who may come having worked on some other system, but you get them excited about coccidioides, they may continue in it. Sometimes easier than a student, because a student goes off and does a postdoc and gets excited about something else. And also where it says individual projects, I think that's fine, but I think those projects need to be collaborative. That don't fund just an individual, but fund a group of people that have assembled the expertise to really address one of these important questions that are in the first set of questions.
Yeah, I want to echo that. I think that if nothing else, what came out of this webinar is the interest across multiple disciplines in addressing this, and I think the strength will come from encouraging people to work together in groups.
Any other comments from the rest of the panels that want to speak to that?
I think training, as Royce pointed out, not just training for scientists, but also training for clinicians. That's a very important thing.
Thank you. So we have time probably for one or two more questions. Did you want to ask one, Uta?
Sure. Actually, earlier I saw, give me a moment.
While you're looking, this is Duke, can I just mention, there is another collaborative research I forgot to mention, but we are open for that. So we are working with Justin at UC Berkeley on a project called looking at the effects of drought and climate change on public health in California. So part of that is looking at valley fever and how this has been affected. So just want to mention that we are collaborating.
Yes, and I am pleased to say that is another one of our system-wide funded programs through our multi-campus program. We are really pleased that we are already making some resources available around this disease area, and we know that there are some multi-campus collaborations on that project, on the one that Dr. Sill is the lead for, and so we're really trying to think about how this extra $3 million that we know that we have at least this year can position our faculty to take the next steps in these important research areas, but also to sort of be well positioned if this forward legislation that Mr. Braden Murphy told us about comes through, how we would be well positioned as a university and as the faculty at the University of California and of course across the state can be in a good position to then leverage and really move forward. Especially he mentioned the matching funds, and so that we see as a real opportunity as well. Did you have a question, Rosa?
Well, so maybe just also, to start wrapping up, and I did want to acknowledge that there was an online discussion around dust science, and that was just very interesting to me, for not myself being a specialist at all in cocci research. And I guess it really speaks to the multidisciplinarity that is needed for such a complex disease, where you have a pathogen and a host, and how the pathogen gets around. I just wanted to acknowledge, and Dr. Taylor already answered a question around the need, around understanding the role of dust particles in the dissemination of the organism. And there was a question whether there was any anecdotal evidence whether infections are primarily due to inhaling the hyphae that are attached to dust, or are they floating on their own? So if there's a very quick answer to that, we are coming to the end of the webinar. I welcome that.
Well, we know that the conidia are infective, so it's very likely that the spores are what is transmitting the disease. That doesn't mean a hypha that had an intact cell couldn't also cause the disease. But my guess is it's the conidia that are most important. Although when you do these environmental sampling, you can't separate the two because you're just getting DNA from nuclei in general. And so you'll get them from fragments of hyphae as well as spores.
And
thank you, Dr. Sill.
I just want to quickly say what fungi do is build spores that are meant to aerosolize. That's how the organisms disperse, pathogens or non-pathogens. So it's very likely that, I mean, maybe dust contributes, but I think the organism can spread on its own.
Thank you. Quickly, Dr. Vujja, quick comment?
Yes, there were 2 studies that actually document people exposed to dust actually came down with it more than at-risk. So, for example, in the early 1990s, there was an earthquake in LA. In one particular area, LA, there were an outbreak and people were exposed to that. Yes. And then the other one was a recent outbreak among construction workers of a solar farm. And definitely when we did the EPI study, exposure to dust and dusty air were clearly a risk factor. So there is an association with dust as well.
Thank you, Dr. Vujja. And I want to take a moment to thank all of our panelists today and all of our attendees for your questions. We are now going to invite our Executive Director Bart Aoki and Vice President Art Ellis back to this chair, the hot seat in the front of the room here, to make some final remarks to everybody. So thank you.
Hi, everyone. I'd like to thank you again to all the participants and all the attendees, for the speakers for taking your time to speak to us today, and to our participants who've attended and contributed to the question and answer session. Again, if we were unable to address your question during the allocated time, please provide your comments and questions to ucri@ucop.edu. Also, please be on the lookout for the post-webinar follow-up survey, which will be sent to you within the next 48 hours. Within the next 2 months, we'll be synthesizing your input, and as I mentioned earlier, developing an RFP that will be issued on and around the early part of December. In the meantime, the recording of today's session will be made available. It will be on our website. And we will also be looking into the distribution of an email list of all of the participants and as well as the both speakers and attendees in order to help in the collaborative process that we've all been talking and discussing about today. Again, an announcement regarding the UC Valley Fever Research Funding Opportunity again will be made available, we anticipate in early December. And at this point, I'd like to turn this back over to Vice President Ellis.
And thanks very much, Bart, and let me add my thanks as well to our speakers and discussants today. This has been extremely illuminating. And as I think all of you know, great advances often come from this combination of bottom up and top down in terms of thinking about ways that the research enterprise can move forward, can integrate with the clinical elements and with the community that we want to serve with the kind of research we're supporting. So I'd like to conclude also by thanking folks who helped put the webinar together, Bart, Kathleen Irwin, who you've met, Uta Grieshammer, who you've also met, Tyler Martz, Jessica Wu, and our Research Grants Program Office, Emily Rader, and here from UC San Francisco, Brian Smith, Irene Broderick, and Benjamin Wallen. So thank you all, and we look forward to staying in touch. If you have further thoughts, please get in touch with us. So many thanks. That concludes our webinar.
Yeah, it does.